Now next chapter is the chapter number 3 hypersensitivity. इसमें हम पढ़ेंगे टाइप 1 हाइपरसेंसिटिविटी, टाइप 2 हाइपरसेंसिटिविटी, टाइप 3 हाइपरसेंसिटिविटी, टाइप 4 हाइपरसेंसिटिविटी. ओके, तो सबसे पहले hypersensitivity. It is known that the adaptive immune response is a significant constituent of host defense against infection and hence essential for maintaining the normal health. The immune response in the form of either cell mediated or humoral mediated, sometimes elicited by antigens not associated with infectious agent, are inappropriate, and this may lead to causation of serious disease. The inappropriate immune response, rather than providing exemption or safety to the affected animal, can produce severe or occasionally fatal results. They are known as hypersensitivity reactions, and the disease caused are known as hypersensitivity diseases. Coombs and Gell, Gell, Coombs, Coombs. Coombs and Gell in 1963 defined 4 types of hypersensitivity based on immunologic mechanism involved. The first 3 types, first to third, are immunoglobulin dependent reaction involving humoral B lymphocytes mediated response. Conversely, type 4 is a T cell mediated hypersensitivity reaction. Overreaction to antigen is also described as allergy, the term originally coined by von Pirquet, Pirquet. एलर्जी टर्म दिया था वॉन पिरक्विट ने. और टाइप फर्स्ट से थर्ड हाइपरसेंसिटिविटी जो होती है वो ह्यूमरल मीडिएटेड होती है, जबकि टाइप 4 जो होती है वो सेल मीडिएटेड होती है. एलर्जी इज डिफाइंड एज द altered reactivity of an animal following exposure to the foreign antigen and included both the immunity and hypersensitivity. The term, however, is now restricted to refer only to hypersensitivity, which may be associated with the development of the immune response to the foreign substance. The essential basis of this hypersensitivity reaction is briefly summarized here, and then each will be described separately in greater detail. Allergic reaction occurs when an already sensitized animal is re-exposed to same allergen. These responses may range from a localized reaction to systemic anaphylaxis, which may even result into death. Allergic response do not occur when a native animal is first exposed to allergen. Instead, initial responses takes time to produce a specific antibody or T cell that react with the allergen, and usually does not produce any symptoms. Any further re-exposure of such animal will produce symptoms. Immune response to allergen, allergen को हम innocuous substances भी कहते हैं. innocuous substances, INNOCUOUS, innocuous substances. तो immune responses to allergens यानी कि innocuous substances are identical to the offending pathogen. In allergic reaction, foreign substances are non-harmful, thus the pathology induces is purely the result of immune response. Hypersensitivity reaction thus demonstrate that immune response itself can cause significant pathology, as it does in some responses to infection. इसमें एक बहुत अच्छी टेबल दी हुई है टाइप फर्स्ट, टाइप सेकंड, टाइप थर्ड एंड टाइप 4 की. The type of hypersensitivity reaction depends on the nature of antigen and its immune responses. The antibody mediated hypersensitivity can be classified into 3 classes. Most common allergies occur due to production of IgE and are kept in type 1 hypersensitivity. This is regarded as classical hypersensitivity reaction, which occurs within seconds or minutes of the antigen contact. IgE antibody bounds mast cell or basophils and activated to release vasoactive amines, leukotrienes and cytokines in the same fashion as the mast cell responds to an infection. Some hypersensitivity reactions are mediated by antibody isotype other than IgE. When the allergen is cell-bound, the antibody activates complement system and Fc mediated effector reaction, and the cell attack in the same way as bacteria. This type of reaction is classified as class 2 hypersensitivity. When the allergen is soluble, immune complexes formed at the site of antigen deposition induce local inflammation and is classified under class 3 hypersensitivity reaction. While type 1 reaction occurs within minute of allergen contact, type 2 and type 3 reaction are observed within a few hours of exposure. In type 4 reaction, the allergen may be a foreign protein or a chemical substance that react with the self protein. Once the animal is sensitized to a modified protein, re-exposure with the same antigen leads to T cell response that evolves over several days. Hence, these reactions are also called delayed type of hypersensitivity. Each reaction occurs in infection with the intracellular bacteria and after contact with certain chemicals. यानी कि इसको हम कॉन्टैक्ट हाइपरसेंसिटिविटी रिएक्शन भी कहते हैं. Thus, it is evident from the foregoing that the class of antibody or T cell produced in response to an allergen determines the type of a hypersensitivity reaction and that will occur following its re-exposure. अब टाइप फर्स्ट हाइपरसेंसिटिविटी है. टाइप फर्स्ट हाइपरसेंसिटिविटी आर एक्यूट इन्फ्लेमेटरी रिएक्शंस that occur as a result of release of biochemically active substances from activated mast cells and basophils. The reaction is mediated by a cytotropic IgE antibody bound to these cells. Antigen that stimulate allergy is called as allergen. An allergen generates a plasma cell and memory cell that secrete IgE. This class of antibody bind with a high affinity of Fc receptors on the surface of mast cell and basophils. The IgE bound mast cells and basophils are referred to as sensitized. Subsequent exposure to the same allergen cross-linked to the membrane-bound IgE on sensitized mast cells and basophils cause their degranulation. The allergy reaction occur immediately after an antigen contact, hence called as immediate hypersensitivity. The reaction may be either systemic or severe, that is anaphylaxis, or localized to certain organ or tissues. The IgE antibody which mediates type 1 hypersensitivity is sensitized predominantly in lymphoid tissues associated with the skin, gastrointestinal and respiratory tract. Most allergic IgE responses occur on mucosal surface in response to allergen that enter the body either by inhalation or ingestion. These allergens includes protein from parasites, helminths, insects, foreign serum, vaccine, plant pollens, drugs, food, mold or spore, etc. The production of IgE against parasitic antigens suggests that IgE evolved specifically to produce resistance against parasite. The self-cure phenomenon is the best example of the beneficial effect of type 1 hypersensitivity. A normal individual produces only small amount of IgE, but some individuals who produces high level of IgE is said to be atopic. The condition is called as atopy, ATOPY. तो जब नॉर्मल इंडिविजुअल कम अमाउंट में IgE प्रोड्यूस करते हैं, बट कभी कभी क्या होता है इंडिविजुअल बहुत हायर लेवल का IgE प्रोड्यूस कर लेते हैं. इसको हम एटोपी कहते हैं, एटोपिक कहते हैं ऐसे इंडिविजुअल को, और इस कंडीशन को हम एटोपी कहते हैं. कंपोनेंट ऑफ टाइप 1 हाइपरसेंसिटिविटी, इसमें सबसे पहले आती है मास्ट सेल. दूसरा आता है कंपोनेंट इम्यूनोग्लोबुलिन E यानी कि IgE, ठीक है. और तीसरा कंपोनेंट आता है IgE बाइंडिंग Fc रिसेप्टर्स, तो Fc रिसेप्टर्स भी आते हैं. चौथा कंपोनेंट आता है मॉलिक्यूलर मैकेनिज्म ऑफ IgE मीडिएटेड डीग्रेनुलेशन, तो ये हम मॉलिक्यूलर मैकेनिज्म पढ़ेंगे. और पांचवा कंपोनेंट आता है इंट्रासेलुलर इवेंट. तो ये 5 कंपोनेंट है इसके बारे में हम पढ़ेंगे. पहला है मास्ट सेल एंड बेसोफिल्स. मास्ट सेल्स आर लार्ज सैटेलाइट और राउंड सेल्स विथ large stellate or round cells with a diameter of 15 to 20 micrometer. They are widely distributed in connective tissue closely associated with blood vessels and lymphatics. It possesses a bean or oval shaped nucleus and contain numerous granules with eccentric distribution. The liver in dog and mesentery in chickens have large number of mast cells. The blood basophils through present in extremely small numbers, that is 0.5 to 2% only, 0.5 to 2% only, plays very important function in the body. It is round shaped with a diameter of 10 to 15 micrometer and has multilobulated nucleus and relatively evenly distributed less numerous granules. Some of the morphological characters such as number, size and stability of granules vary amongst domestic species. It is believed that basophils are derived from promyelocytes in the bone marrow. Morphologically, it is resembled with the mast cell and shares similar function. Granules of mast cell and basophils stain metachromatically at low pH with certain dyes such as Alcian blue and toluidine blue, etc. Alcian, ALCIAN, Alcian blue और toluidine, TOLUIDINE, toluidine blue से भी ये स्टेन हो सकता है. Significant variation has been noted in the mast cell population at different sites with regard to type and amount of allergy mediators that contain and their sensitivities to activating stimuli and cytokines. Precursors of mast cell are formed in the bone marrow and are carried to virtually all vascularized peripheral tissues where they differentiate into mature cells. Two types of mast cells have been identified in the rodents and humans. One type is found in the connective tissue and the other is associated with the mucosal surface. Connective tissue mast cells contain many uniform granules and are rich in histamine and heparin. On the contrary, mucosal associated mast cells have few variable sized granules that contain chondroitin sulfate, not heparin. इसमें हेपेरिन नहीं होता, chondroitin sulfate होता है. They contain little histamine and produce different prostaglandin and leukotrienes as well as platelet activating factor PAF. Mucosal mast cells can proliferate in response to interleukin 3 and interleukin 4 which are secreted from T cell. They are also activated in response to parasitic Helminth's antigen. Mast cell and basophils possess a variety of biological activity performed mediators and are capable of synthesizing several other pharmacologically active substances upon immunologic and non-immunologic stimulations. These mediators are released in a sequential fashion and are important mediators of allergy and other inflammatory catecholamines and other inflammatory reactions. Both the cells have receptors for IgE, IgG, beta adrenergic binding, beta adrenergic catecholamines and prostaglandins and histamines. In comparison to IgE, IgG binding is less effective in inducing degranulation. Cell surface receptor density for IgE is more on the basophil, that is 15,000 से 6 lakh receptors per cell होते हैं इसमें, then on the mast cell में 3,000 से 6 lakh receptors होते हैं. Comparative features of mast cell and basophils are summarized in table 3.2. तो इसको आप टेबल 3.2 में देख सकते हैं कि इन दोनों में क्या फीचर्स हैं, कंपैरेटिव फीचर्स क्या क्या हैं. दूसरा कंपोनेंट आता है इम्यूनोग्लोबुलिन IgE. C.R. Richard finding for the first time suggested that some constituent of serum was responsible for hypersensitivity in dog. Systemic anaphylaxis could be produced in a unsensitized dog by injecting serum from a sensitized dog. Similar component in the human serum was demonstrated that was first later identified as IgE antibodies. It is found in the serum in very small quantity, 9 to 700 microgram per ml in the dog, which made its physiochemical characterization difficult. Discovery of an IgE myeloma in 1967 leads to comprehensive chemical analysis of IgE. It is heat labile immunoglobulin and composed of 2 heavy and 2 light chain with a molecular weight of 1,90,000. The increase in molecular weight is due to an additional conformation of Fc portion of molecule. It enables IgE to bind to glycoprotein receptor on the surface of mast cell and basophils. The half-life of IgE in the serum is only 2 to 3 days, but when bound to the mast cell, it is stable for few weeks. The serum IgE level is found to be related with parasite burden of the animal. Though affinity of IgG1 to mast cell is extremely low, it has been found to mediate atopic dermatitis in dogs. IgE binding Fc receptors, ये तीसरा कंपोनेंट है. The reagenic activity, REAGINIC. The reagenic activity of IgE is dependent upon its ability to bind to receptors specific for the Fc portion of heavy chain. Two types of receptor have been identified as a high affinity Fc epsilon Ri and a low affinity यानी कि FCER second, मतलब epsilon FC epsilon R first और FCER second. CD23 receptors. They are expressed by different type of cells including mast cells, basophils and eosinophils. The affinity of Fc epsilon R first for IgE is 1000 times more than Fc epsilon R second, which enables it to bind IgE almost irreversibly, deposit very low concentration of IgE in the serum. The Fc epsilon Ri receptor consists of 4 polypeptide chain in alpha and beta chain and 2 gamma chains linked with disulfide bond. The extra cytoplasmic portion of alpha chain binds with the methyl CH3 domain of IgE molecule via its 2 immunoglobulin like molecules. The beta chain acts as an linker between alpha chain and gamma homodimers. The 2 gamma chain extend deeper into the cytoplasm and contain immunoreceptors tyrosine activation motif यानी कि ITAM, which interacts with the protein tyrosine kinase PTK. PTK is responsible for tyrosine phosphorylation and this initiates the process of mast cell degranulation. The FC epsilon R second यानी कि इसका दूसरा नाम है CD23 is another receptor with lower affinity for IgE in the comparison of Fc epsilon R first and thus belongs to immunoglobulin superfamily. The FCER second receptor plays a variety of function in regulating the intensity of IgE responses. It is found on NK cells, eosinophils, platelets, macrophages, dendritic cells and on nearly 1/3 population of B cells. Molecular mechanism of IgE mediated degranulation. ये next component है. The mechanism leading to degranulation remains same for both the mast cell and the basophil. The first step is to binding of allergen to the IgE and cross-linking the reaction. The cross-linking the receptors bound IgE on the surface of basophil and mast cell. The binding of IgE alone on the mast cell does not trigger the degranulation process. Thus, monovalent allergen cannot initiate the process. Experimental studies have shown that cross-linkage of two or more Fc epsilon or first receptor are necessary for degranulation to occur, which can be achieved by binding with allergen or other cross-linking method, that is the anti-IgE antibody, anti-receptor antibody, and anti-idiotypic antibody. It has also been seen that ratio of IgE to allergen should be 2:1 or more to induce degranulation. Next component है intracellular event. Intracellular event consists of several sequential steps involving receptor cross-linkage, activation of protein tyrosine kinase, methylation of membrane phospholipids, influx of calcium ions, change in the cyclic AMP level, and the fusion of granules with the plasma membrane. Cross-linkage of IgE by allergen results into aggregation of Fc epsilon or first receptor and the activation of PTK associated with the cytoplasmic domain of beta and gamma chain. PTK subsequently phosphorylates phospholipase C, which convert phosphatidyl inositol 4,5 biphosphate into diacyl glycerol DAG and inositol triphosphate. This in turn increase intracellular calcium level and activate protein kinase PKC. Activated PKC phosphorylates myosin light chain necessary for microtubular assembly and fusion of granules with the plasma membrane and releasing their content into extracellular fluid. Simultaneously, Fc epsilon or first cross linkage also activates phospholipase, which acts on membrane phospholipid to produce arachidonic acid. Two classes of potent mediator are produced from arachidonic acid. The पहला है prostaglandins and दूसरा है leukotrienes. Finally, the protein kinase promote translation and expression of gene encoding several cytokines such as interleukin 4, interleukin 5, interleukin 6, and tumor necrotic factor alpha, that is TNF alpha. Degranulation of mast cell in basophils occurs within seconds of binding of allergen to IgE on the cell surface. Degranulated mast cells do not die, but their identification becomes difficult due to changed morphology. तो ये था अभी तक इसके कंपोनेंट. अब आगे हम पढ़ेंगे मीडिएटर्स ऑफ टाइप फर्स्ट हाइपरसेंसिटिविटी रिएक्शन. Now comes to the mediators of type 1st hypersensitivity reaction. Following cross binding of Fc epsilon R1 receptor by allergen on the surface of the mast cell, several pharmacologically active agents are released in the extracellular fluid. The clinical manifestation of type 1st hypersensitivity in the fact results from biological effect of these agents, which can be classified into primary and secondary mediators. The primary mediators are those which are produced before degranulation and stored in granules. This includes histamine, serotonin, proteases, chemotactic factor for eosinophil and neutrophil, and heparin. The secondary mediators are sensitized following activation of mast cell and includes platelet activating factor, leukotrienes, prostaglandins, bradykinins, and various cytokines. The difference in clinical manifestation of the type of reaction amongst different species of animals may partly reflect variation in the primary and secondary mediator secretions. अब इसके बाद नेक्स्ट है प्राइमरी मीडिएटर्स। ये कौन कौन से होते हैं? हिस्टामिन एंड सेरोटोनिन आर द मोस्ट इंपॉर्टेंट वेसोएक्टिव एमीन्स विच आर फॉर्म्ड इन द हाई कंसंट्रेशन इन द ग्रेन्यूल्स ऑफ mast cell. Its biological effect are observed within minutes of mast cell activation. Histamine exerts its effect by binding with H1 receptor present on different tissues. It includes contraction of smooth muscle in bronchi, gastrointestinal tract, uterus and bladder, and increased permeability of vessels. Histamine stimulates mucus secretion by goblet cell, lacrimation and salivation. ट्रिप्टोफेन डेरिवेटिव सेरोटोनिन यानी कि 5-हाइड्रोक्सीट्रिप्टामीन is released from mast cell of rodents and large ruminants. The main function of vasoconstriction that results in rise in the blood pressure. In cattle, however, serotonin is a vasodilator. It has only minor effect on vascular permeability except in the rats and mice. Other mediators which are rapidly released from mast cells include eosinophilic chemotactic factor (ECF) and neutrophilic chemotactic factor यानी NCF. Infiltration of eosinophils is often related with the type 1 reaction. In addition to this, more than 50% of protein content of granules are made up of neutral proteases such as trypsin and chymotrypsin. The complement components C3 and C5 are cleaved by these proteases to generate anaphylactoxin यानी कि C3A and C5A. The mast cell granules also contain kallikreins which act on kininogen to produce kinins. Secondary mediators. This includes lipid mediators and cytokines. Lipid mediators prostaglandins and leukotrienes are produced as a result of enzymatic cascade. Activated phospholipase acts on the membrane of phospholipids to produce arachidonic acid. The latter leads to production of prostaglandins, prostacyclins, and thromboxanes via cyclooxygenase pathway. Prostaglandin causes intense bronchospasm and increased mucus secretion. Leukotrienes are potent vasoactive and spasmogenic agent. They increase vascular permeability and cause bronchial smooth muscle contraction several thousands times more than histamine. Leukotrienes यानी कि B4, C4 and D4 were earlier collectively called as slow releasing substances of anaphylaxis यानी कि SRSA. In addition, leukotrienes B4 is highly chemotactic for neutrophils, eosinophils and monocytes. Next is cytokines. Mast cell and basophils secrete cytokines which further complicate the inflammatory process. Some of these cytokines are involved in clinical manifestation of type 1 hypersensitivity reaction. Interleukin 4, interleukin 5, interleukin 6, interleukin 13 and tumor necrotic factor alpha change the local microenvironment leading to recruitment of several inflammatory cells such as neutrophil and basophils. Clinical manifestations. अब आते हैं अगला. The consequence of type 1 reaction in an animal depends on several factors including genetic constitution of the animal, nature and amount of allergen, tissue distribution of mast cell and its content in the target organ, and most importantly the route of administration of the allergen. The clinical manifestation may range from serious life-threatening conditions such as acute systemic anaphylaxis to localized allergic reaction of limited consequences. अब इसमें पहला पढ़ते हैं acute systemic anaphylaxis. Systemic anaphylaxis occur within minutes of type 1 hypersensitivity reaction and usually fatal if not attended. This type of reaction was observed for the first time in 1839 in dog following repeated injection of egg albumin. The importance of the finding was, however, realized only in 1902 when two French physicians were attempting to develop antitoxin to protect swimmers from painful stings of jellyfish and sea anemones. They found that after primary immunization of dog with sublethal dose of extract of C. anemone, second injection after several weeks caused rapid development of symptoms like vomiting, bloody diarrhea, asphyxia, unconsciousness, and death. They named this phenomenon as anaphylaxis. तो anaphylaxis नाम जो था वो दो साइंटिस्ट ने दिया था. ग्रीक वर्ड है इसका एना यानी कि अगेंस्ट और फाइलेक्सिस यानी प्रोटेक्शन, यानी कि प्रोटेक्शन के अगेंस्ट. ठीक है. इन कंटेस्ट विथ देयर फाइंडिंग्स दे वेयर लुकिंग फॉर ए प्रोफाइलेक्टिक बट इन रिटर्न गॉट एनाफाइलेक्टिक. Clinical and pathological features of acute anaphylaxis differs amongst domestic species and humans and are summarized in table 3.4. अब केटल में बात करते हैं. मेजर मीडिएटर ऑफ एनाफाइलेक्सिस इन द केटल इज सेरोटोनिन, काइनिन्स एंड द ल्यूकोट्राइन्स. हिस्टामिन इज ऑफ लिटिल सिग्निफिकेंस. The main shock organ is the lungs, which shows hypertension due to constriction of pulmonary veins resulting in dyspnea and pulmonary edema. Hypersecretion of mucus further aggravates the respiratory difficulty. Affected animals show urination, defecation, and blotting, which results from smooth muscle contraction of urinary bladder and intestine. Dopamine enhances histamine and leukotriene secretion from the bovine lungs in the acute anaphylaxis. The alpha and beta adrenoreceptors on the mast cell of cattle behave differently in comparison of the other species. The drug that stimulates beta receptor potentiate histamine release from leukocytes, whereas the drug that stimulates beta receptor inhibit histamine release. The mechanism of this peculiar effect is not yet clear. ये था केटल में. शीप में बात करें तो acute anaphylaxis in the sheep presents almost similar symptoms as seen in the cattle. Pulmonary signs predominate as a result of constriction of bronchi and blood vessel. Smooth musculature of intestine and urinary bladder is also affected with expected results. हिस्टामिन इज़ द मेजर मीडिएटर बिसाइड्स सेरोटोनिन, ल्यूकोट्राइन्स एंड काइनिन्स. तो हिस्टामिन जो होता है मेजर मीडिएटर होता है शीप में. अब हॉर्स में बात करते हैं. इन इक्वाइन्स लंग्स एंड इंटेस्टाइन आर द मेन ऑर्गन्स टू बी अफेक्टेड इन द सिस्टेमिक एनाफाइलेक्सिस. क्लिनिकल साइन कंसिस्ट ऑफ कफ, डिस्पनिया leading to apnea and the consequence of the histamine and serotonin mediated bronchial and bronchiolar constriction. तो इंटेस्टाइन और लंग सबसे ज़्यादा अफेक्टेड होते हैं हॉर्स में, और इसमें डिस्पनिया होता है, एपनिया भी हो जाता है और ब्रोंकियल कॉन्स्ट्रिक्शन हो जाता है. इन द पोस्टमार्टम एग्जामिनेशन पल्मोनरी एम्फाइसीमा एंड पेरी ब्रोंकियल एडिमा आर कंसिस्टेंटली सीन. द एनिमल में ऑल्सो शो डायरिया एंड हेमोरेजिक एंट्रोकोलाइटिस. हॉर्स हो गया. हॉर्स के बाद में पिग आते हैं. इन पिग ऑल्सो द मेजर शॉक ऑर्गन इज द लंग्स एंड द प्रिंसिपल फीचर ऑफ एक्यूट एनाफाइलेक्सिस इंक्लूड सिस्टेमिक एंड पल्मोनरी हाइपरटेंशन. तो इसमें भी शॉक का जो मेन ऑर्गन होता है वो लंग्स होता है, और जो इसका जो मेन फीचर है वो है सिस्टेमिक एंड पल्मोनरी हाइपरटेंशन होने के कारण होता है. विच आर मोस्टली हिस्टामिन मीडिएटेड. हिस्टामिन के कारण ही होता है. द एनिमल डाई ड्यू टू रिस्पायरेटरी डिस्ट्रेस. इंटेस्टाइन्स आर इनफ्रिक्वेंटली इन्वॉल्व. तो इंटेस्टाइन कभी कभारी मतलब इंटेस्टाइन जो होता है नॉर्मली इन्वॉल्व रहती है, और एनिमल जो होता है उसकी डेथ रिस्पायरेटरी डिस्ट्रेस से होती है. डॉग्स एंड कैट्स. Acute anaphylaxis in the dogs, particularly in respect of the shock organ, differs from those of domestic species. Hepatic veins in the liver is a major site of action for mediators such as histamine, prostaglandins, and leukotrienes. Clinical symptoms in the anaphylactic dog include initial excitement, followed by vomiting, defecation, and urination. With the process of the reaction, dog shows muscular weakness, muscular weakness and depressed respiration, convulsions, and becomes comatose and die within 45 to 60 minutes. तो डॉग्स के केस में भी जो शॉक ऑर्गन होता है वो डिफरेंट होता है अदर स्पीशीज़ से। और यहाँ पर जो हिपेटिक वेन है जो लीवर में जाती है वो वहाँ पर मेन साइट होता है जहाँ पे हिस्टामिन अपना एक्शन करता है, प्रोस्टाग्लैंडिन अपना एक्शन करता है, ल्यूकोट्राइन्स अपना एक्शन करता है। और डॉग्स में नॉर्मली इसके बाद एक्साइटमेंट होता है, वोमिटिंग होता है, डेफिकेशन और यूरिनेशन होता है। इसके बाद मसल वीकनेस आती है, रिस्पायरेशन लो हो जाता है, कन्वल्जन्स आते हैं और 45 से 60 मिनट्स में डेथ हो जाती है। Postmortem examination reveals a severe engorgement of the liver and intestine holding more than 60% of total blood volume. These occur as a result of hepatic vein occlusion due to smooth muscle contraction and swelling of hepatic cell. तो इस जब पोस्टमार्टम करते हैं तब पता चलता है कि 60% जो ब्लड वॉल्यूम है वो सिर्फ हेपेटिक वेंस में है और इसके कारण लीवर का इंगॉर्जमेंट हो जाता है और इंटेस्टाइन का इंगॉर्जमेंट हो जाता है। और ऐसा बिकॉज हेपेटिक सेल की स्वेलिंग हो जाती है, ऐसा बिकॉज ऑफ मसल कॉन्ट्रैक्शन के कारण होता है। केट्स में बात करें तो द केट्स द मेजर शॉक ऑर्गन इज द लंग्स एंड मीडिएटर्स आर एनाफाइलैक्सिस आर हिस्टामिन एंड ल्यूकोट्राइन्स। केट्स में लंग्स होता है जहाँ पे शॉक होता है, इसके अलावा हिस्टामिन और ल्यूकोट्राइन्स मेजर एनाफाइलेक्टिक एजेंट्स हैं। The animal shows scratching of faces and heads, dyspnea, salivation, vomiting, incoordination and collapse. Pulmonary emphysema, hemorrhages and edema, bronchial constriction and edema of glottis are significant lesion observed at necropsy. तो कैट्स की कंडीशन में मेन ऑर्गन लंग्स है जहाँ शॉक होता है और मीडिएटर्स जो है वो हिस्टामिन है और हिस्टामिन के अलावा ल्यूकोट्राइन्स हैं। और एनिमल में जो सिम्टम्स मिलते हैं वो स्क्रैचिंग, फेस का स्क्रैचिंग करेगा, हेड का स्क्रैचिंग करेगा, डिस्पनिया करेगा। डिस्पनिया होने के बाद बहुत ज़्यादा स्लाइवेशन होगा, वोमिटिंग होगी, इनकोऑर्डिनेशन होगा और कोलैप्स हो जाएगा। नेक्रोप्सी में पल्मोनरी एम्फाइसीमा मिलता है, हेमोरेजेस मिलते हैं एंड एडिमा मिलता है एंड ब्रोंकियल कंस्ट्रक्शन मिलता है एंड एडिमा ऑफ ग्लॉटिस, मेन है कि ग्लॉटिस का एडिमा मिलता है। अब नेक्स्ट है लोकलाइज्ड स्पेसिफिक एलर्जिक कंडीशंस। Localized allergic condition are most commonly seen that the acute anaphylaxis. Organs affected reflects the route of entry of allergens. For example, hay fever results from fluid exudation from nasal mucosa as inhaled antigen acts on upper respiratory tract, trachea and bronchi. तो hay fever में नॉर्मली एक्चुअल में ऑर्गन जो है डिपेंड करता है कि कौन सा शॉक लगा है, उसके लिए एलर्जन कहाँ से एंटर हुआ है, इस पर डिपेंड करता है। तो जैसे मान लीजिए है फीवर के केस में नेज़ल म्यूकोज़ा में बहुत ज़्यादा एक्सुडेशन मिलेगा, अपर रेस्पिरेटरी ट्रैक्ट में क्योंकि यहाँ और ट्रेकिया और ब्रोंकाई में यहाँ पर एंटीजन गया हुआ है। अस्थमा इन द ह्यूमन्स अकर ड्यू टू ट्रेकियोब्रोंकुलर कंस्ट्रक्शन इन रिस्पॉन्स टू द इनहेल्ड एंटीजन्स। सिमिलरली एलर्जिक कंजक्टिवाइटिस, articaria, diarrhea and hemorrhagic enteritis may occur as a result of antigen contact to respective organs. Following are some of the species of the specific localized allergic condition in the animal. सबसे पहले मिल्क एलर्जी की बात करें तो ये डिपेंड करता है कि शॉक जिस ऑर्गन में लगा है वो किस ऑर्गन में एलर्जन गया है। जैसे है फीवर के केस में जो एलर्जन होता है वो रिस्पायरेटरी ट्रैक्ट में जाता है, अस्थमा के केस में भी रिस्पायरेटरी ट्रैक्ट में ह्यूमंस में जाता है। तो अब सबसे पहले बात करते हैं मिल्क एलर्जी की। सर्टेन ब्रीड्स ऑफ हाई यील्डिंग काउज सच एज जर्सी माउंट्स इम्यून रिस्पॉन्स टू इट्स ओवन अल्फा केसिन ऑफ मिल्क which is sensitized in the udder. This protein does not get access to blood circulation if the cow is milked regularly. That avoids increased inflammatory pressure. Intramammary pressure. Delay in the milking causes undue increase in the intramammary pressure, which allows seepage of milk protein in the circulation and produce the cytotropic IgE antibody. Subsequent release of alpha casein proteins results in type 1 reaction, which may vary from mild discomfort with arterial skin lesions to acute and systemic anaphylaxis and death. Regular milking prevents this occurrence of such condition in the high yielding cows. तो मिल्क एलर्जी सबसे ज़्यादा काउज में देखने को मिलती है। अगर उनका रेगुलर मिल्किंग नहीं होता तो उस कंडीशन में जो जर्सी है उस जर्सी में ये देखने को मिलता है कि मिल्क में अल्फ़ा केसिन प्रोटीन होता है, और मिल्क का अल्फ़ा केसिन प्रोटीन अगर रेगुलर मिल्किंग करते रहें तो वो ब्लड में मिक्स नहीं होता, लेकिन जब इंट्रामेमरी प्रेशर बढ़ जाता है क्योंकि आपने इधर मिल्किंग नहीं किया, तो इंट्रामेमरी प्रेशर बढ़ने के कारण वो अल्फ़ा केसिन जो है ब्लड में जाता है, और ब्लड में जाने से वहाँ पर टाइप फर्स्ट हाइपर सेंसिटिव रिएक्शन होता है, आईजीई प्रोडक्शन होता है, और आईजीई प्रोडक्शन होने के बाद में एनाफाइलेक्टिक शॉक लगता है और डेथ हो जाती है। तो इसलिए रेगुलर मिल्किंग बहुत ज़रूरी है। नेक्स्ट है फूड एलर्जी। Food allergy in the dog and cat is common condition and causes allergic dermatitis and gastrointestinal disorder. Protein-rich foods such as milk and milk products, wheat meal, fish, chicken, beef, and egg may cause allergy. About 15% of dogs suffering from food allergy shows gastrointestinal involvement. The clinical sign involves vomiting, cramps, loose, fetid stool, and in the several cases, hemorrhagic diarrhea may be seen as soon after the intake of food. Skin lesions in the food allergy have been found in about 50% of the affected dogs. Eyes, ears, feet, axilla, and perineal area are usually involved and shows reactions consisting of erythematous papillae. The lesions are highly pruritic because of histamine and are frequently complicated with the self-inflicted injury and secondary bacterial fungal infection. In chronic cases, skin is further thickened, hyperpigmented, and secondarily infected. In pigs, the fish meal and alfalfa, and in the horses, wild oats, white clover, and alfalfa have been incriminated as potential food allergens. Treatment of food allergy includes correct identification of food responsible for allergy and replacing with the hypoallergenic diet. Dietary elimination of the food component one by one is the best mean of the diagnosis. Intradermal skin testing is not very much useful in detecting allergy to food component or ingredient. तो फूड एलर्जी बहुत सारे एनिमल्स में मिलती है। डॉग के केस में देखें तो डॉग और कैट के केस में जो डर्मेटाइटिस होता है और जो गैस्ट्रोइंटेस्टाइनल डिसऑर्डर होता है वो फूड एलर्जी से हो सकता है। लगभग 15% डॉग्स को इस टाइप से एलर्जी होती है। कुछ मिल्क प्रोडक्ट्स हैं, मिल्क हैं, कुछ फिश हैं, चिकन है, बीफ है, एग है, व्हीट मील है, इससे एलर्जी होती है। उसमें वोमिटिंग होती है, क्रैम्प होते हैं, लूज फटेड स्टूल होता है और कभी कभी हेमोरेजिक डायरिया भी हो जाता है। ठीक है, इसके अलावा इनके आइज़, ईयर, फीट, एक्सिली और पेरिनियल एरिया में डर्मेटाइटिस होता है। इरिथमेटस पेप्यूल्स बनते हैं जो फर्दर सेकेंडरी बैक्टीरियल इन्फेक्शन होने के कारण या फंगल इन्फेक्शन होने के कारण क्रोनिक हो जाते हैं। इसके अलावा हॉर्स के केस में वाइल्ड ओट्स और वाइट क्लोवर और अल्फाल्फा, इससे एलर्जी होती है। इन पिग्स के केस में फ़िश मील और अल्फाल्फा से एलर्जी होती है। नेक्स्ट है रिस्पायरेटरी एलर्जी। Inhalant allergy is the most commonly manifested as atopic dermatitis in the dog and cat. The major inhalant allergens identified are molds, weed, and pollen grains, especially those which are very small, light, and produces in large quantity. Erythema and edema of face, axilla, and foot are observed in uncomplicated cases. However, any part of the body can be affected. Secondary complication leads to crushing, scaling, and hyperpigmentation and pyogenic infection of the skin. Some dogs may also suffer with the otitis externa and conjunctivitis. Allergic rhinitis and conjunctivitis induced by pollens are less frequently observed in inhalant allergy. It is characterized by excessive lacrimation and watery nasal discharge. तो ये एलर्जेंस होते हैं जो डॉग्स के केस में एलर्जी प्रोड्यूस करते हैं रिस्पायरेटरी। इसको एटोपिक डर्मेटाइटिस भी करते हैं डॉग के केस में। इनहेलेंट्स, तो ये इनहेलेंट्स क्या होते हैं? वीड होते हैं, मोल्ड होते हैं, पॉलन ग्रेन्स होते हैं। जब बहुत ज़्यादा क्वांटिटी में प्रोड्यूस होते हैं ये, और इनहेलेशन होता है इसका, तो एरिथमा होता है, एडिमा, फेस का एडिमा होता है, एक्जिला एंड फूट का जो है इसमें भी एडिमा होता है। है ना। कभी कभी कंजक्टिवाइटिस होता है, और कंजक्टिवाइटिस के अलावा ऊटाइटिस भी होता है, ऊटाइटिस एक्सटर्ना। अब इसके बाद आते हैं नेक्स्ट, केटल में क्या होता है। एलर्जिक राइनाइटिस मतलब वही अभी इन्हेलेशन एलर्जेंस पढ़ रहे हैं। तो केटल के केस में होता है एलर्जिक राइनाइटिस अकर्स इन द केटल एज अ रिजल्ट ऑफ इन्हेलेशन ऑफ एंटीजन फ्रॉम नंबर ऑफ प्लांट्स एंड फंजाई। Clinically, the affected animals shows excessive lacrimation, mucoid nasal discharge, and intense pruritus at the nasal region. In chronic cases, nodular growth is formed in the nasal mucosa, which consists of large number of mast cells, eosinophils, and plasma cells. तो इसका भी केटल में भी ये होता है, ठीक है। इक्वाइन्स के केस में क्रोनिक ऑब्स्ट्रक्टिव पल्मोनरी डिसीजेस COPD in horses is analogous to asthma in the humans. It is thought to be the bronchopulmonary mold allergy, which is the involvement of type 1 hypersensitivity in its pathogenesis. As seen in the allergic asthma in the human, the major component of late phase reaction is the leukotrienes, cause sustained smooth muscle contraction. The ineffectiveness of antihistamine in the treatment of this disease suggests the role of late phase reactivity. COPD affected horses exhibit positive skin reaction to extract from several actinomyces and fungi including Microspora faini, Polyspora faini, Aspergillus species, and Cladosporium species. But horses with COPD also show positive skin reaction. Also, there have been no correlation between circulating mast cell mediators and severity of COPD. This indicates that precise etiopathogenesis of the condition is unclear. तो इक्वाइन के केस में एक्चुअल में क्रोनिक ऑब्स्ट्रक्टिव पल्मोनरी डिसीज़ होती है, और ये मेनली जो एलर्जेंस होते हैं, मोल्ड हैं, इसके कारण होती है। इसमें टाइप 1 हाइपरसेंसिटिविटी होती है, और ऐसा ही ह्यूमंस में होता है। ह्यूमंस में भी जब पहले तो वो हिस्टामिन से होती है, लेकिन बाद में वो ल्यूकोट्राइन से होने लगती है। तो जब लेट फेज़ में आप हिस्टामिन देते हैं तो हिस्टामिन का कोई रिएक्शन, हिस्टामिन से कोई आपको बेनिफिट देखने को नहीं मिलता है एंटीहिस्टामिनिक ड्रग देने से, क्योंकि वो लेट फेज़ में ल्यूकोट्राइन से ह्यूमंस में एलर्जी होती है। इसके अलावा हॉर्सेस में स्किन रिएक्शन होते हैं, और स्किन रिएक्शन बहुत सारे फंजाइज से भी होते हैं, जैसे माइक्रोपोलिस्पोरा फेनी से, एस्परजिलस स्पीशीज से, क्लेडोस्पोरियम स्पीशीज से, ठीक है। तो इससे पॉजिटिव स्किन रिएक्शन देखने को मिलता है। एलर्जीज टू पैरासिटिक एंटीजेंस इन एनिमल्स। पैरासाइट की बात करें तो IgE mediated reactions to the parasitic antigens plays a significant role in the resistance to helminth infection. The helminth infection stimulate IgE production, which is responsible for allergy and even anaphylactic reaction. The beneficial role of type 1 hypersensitivity was first observed in the form of self-cure phenomena in the helminth infection. A variety of parasitic infection has been identified to cause type 1 reaction in the animal. Animals affected with cystodes sometimes show symptoms of respiratory distress and urticaria. In dogs, a rupture of hydrated cyst during the surgical manipulation or due to other reasons may cause anaphylaxis. Transfusion of blood from dog infected with the heartworm, जैसे Filaria amitis, is already sensitized dog may result in anaphylaxis. तो देखो एलर्जिक पैरासिटिक एलर्जिक में क्या होता है कि जैसे ये IgE का जो प्रोडक्शन होता है उससे बहुत सारे हेलमिन्स बाहर हो जाते हैं, एक्सपेल्ड आउट हो जाते हैं। इसको हम सेल्फ क्योर फिनोमेना कहते हैं, क्योंकि ये एक एलर्जिक एनाफाइलेक्टिक रिएक्शन है। तो पहली बार में इम्यूनिटी प्रोड्यूस नहीं हो पाती इतनी, सेकेंड टाइम पर जब वापस से उन पैरासाइट का इंट्रेंस होता है और इम्यूनिटी रहती है तो वो सेल्फ क्योर फिनोमेना के अंदर आते हैं। अब इसके अलावा कुछ सिस्टोड जो होते हैं वो रिस्पायरेटरी डिस्ट्रेस कॉज करते हैं और आर्टिकेरिया कॉज करते हैं डॉग में। इसके अलावा हाइड्रेटेड सिस्ट भी कभी कभी रप्चर हो जाते हैं तो उससे एनाफाइलेक्टिक शॉक होता है। इसके अलावा जब डायरोफाइलेरिया इमिटिस जो हार्टवर्म ऑफ डॉग है उसका भी जब ट्रांसफ्यूज़न करते हैं ब्लड का किसी हेल्दी डॉग में, तो वो उस डॉग में भी एनाफाइलेक्टिक रिएक्शन हो जाता है। Antigens from certain arthropod parasite have been shown to cause allergies in the man and animal. Acute anaphylaxis in sensitized human due to insect stings are commonly observed. Cattle infected with the hypoderma bovis, एक वर्बल फ्लाई है, में exhibit anaphylaxis as a result of rupture of pupal stage during its physical movement from the back of animal. Release of pupal coelomic fluid in the sensitized animal may cause acute reaction and occasionally death of the animal. तो जो फिर से पढ़ता हूँ मैं, एंटीजेंस फ्रॉम सर्टेन आर्थ्रोपोड पैरासाइट हैज़ बीन शोन टू कॉज एलर्जीज इन द मैन एंड एनिमल। तो बहुत सारे आर्थ्रोपोड होते हैं, वो काटते हैं तो एलर्जिक रिएक्शन होती है। केटल में हाइपोडर्मा बोविस एक वर्बल फ्लाई है, उससे एलर्जिक शॉक होता है क्योंकि रप्चर ऑफ प्यूपल स्टेज ड्यूरिंग इट्स फिजिकल मूवमेंट फ्रॉम बैक ऑफ द एनिमल। तो वो एनिमल के बैक से मूव करता है तो वो वहाँ पर रप्चर होता है उसका प्यूपल स्टेज, तो वो बहुत ज़्यादा एलर्जिक होता है। और रिलीज ऑफ प्यूपल सिलोमिक फ्लूइड, जो सिलोमिक फ्लूइड है, जब वो एनिमल से सेंसिटाइज होता है तो एनिमल की डेथ भी हो जाती है। बाइटिंग ऑफ कैटल एंड हॉर्सेस बाय सर्टेन इंसेक्ट सच एज मिडगेस यानी कि क्यूलिकोइडिस वगैरह, एंड ब्लैक फ्लाइज यानी कि सिमिलियस causes allergic dermatitis popularly known as गल्फ कोस्ट इच। इसको हम गल्फ कोस्ट इच कहते हैं या फिर क्वींसलैंड इच कहते हैं या स्वीट इच कहते हैं। तो कैटल और हॉर्स में क्या होता है, जो क्यूलिकोइडिस होते हैं या फिर जो सिमिलिस होते हैं, ब्लैक फ्लाइज है ना, मिडगेस यानी कि क्यूलिकोइडिस और ब्लैक फ्लाइज यानी कि सिमिलिस, जब इसका बाइटिंग होता है तो उसके कारण एलर्जिक डर्मेटाइटिस होता है। उसको हम गल्फ कोस्ट इच कहते हैं या फिर क्वींसलैंड इच कहते हैं या फिर स्वीट इच कहते हैं। Saliva associated antigens of this parasite causes formation of urticaria, that is intensely pruritus. Mites can also stimulate IgE production and hence lesions produced by them show features of type 1 hypersensitivity. Mange in the dog and cat caused by Sarcoptera scabi and Octodectus cyanotis respectively are the example. Dermal lesions consist of infiltration with the mast cell, lymphocytes and plasma cells. The affected animals show an immediate heat and flare response following intradermal injection of mite antigen. Type 3 reaction mediated immune complex formation may also be associated with the lesions development. Other helminths parasites such as Ascarisum in the pig and Fasciola hepatica in the calves have been shown to induce type 1 hypersensitivity reaction. Apart from a damaging role, IgE antibodies plays a significant protective role in the several parasitic infection. Now next है, कभी ड्रग और वैक्सीन के कारण भी एलर्जीज होती हैं, उसके बारे में पढ़ते हैं। Some drugs and vaccines elicit IgE response and causes discomfort to man and animal mediated by type 1 hypersensitivity reaction. Among the drugs, penicillin allergy is the most notable one. The animal may be exposed to penicillin either through therapeutic dose or injection of contaminated milk. Contaminated with मतलब जिसमें पहले से ही पेनिसिलिन मिक्स हो, ऐसा मिल्क पीने से। द पेनिसिलिन मॉलिक्यूल इज डिग्रेडेड इनटू सेवरल कंपाउंड इंक्लूडिंग पेनिसिलॉइल, तो पेनिसिलॉइल में कन्वर्ट हो जाता है वो। दिस बाइंड्स टू प्रोटीन इन द बॉडी एंड माउंट IgE रिस्पॉन्स। री एक्सपोजर ऑफ सच सेंसिटाइज्ड एनिमल में लीड्स टू एक्यूट एनाफाइलेक्सिस और माइल्डर फॉर्म ऑफ एलर्जी। Antibodies and hormones may also cause allergies in the domestic animals. The induction of IgE response leading to severe allergies have been reported in the cattle, and with the use of killed FMD vaccine, rabies vaccine and contagious bovine pleuropneumonia vaccine, इससे भी हो जाता है. अब डिटेक्ट कैसे करेंगे टाइप फर्स्ट हाइपरसेंसिटिविटी? द मोस्ट सिंपल मेथड टू असेस द आइडेंटिफाई द टाइप फर्स्ट रिएक्शन इन द इंट्राडर्मल इंजेक्शन टेस्टिंग. तो इंट्राडर्मल टेस्टिंग करेंगे हम सबसे पहले. A small amount of potential and suspected allergen is applied at the specific skin site by intradermal injection or superficial scratching. If the animal is hypersensitive to the allergen, local mast cell will degranulate, and the release of histamine and other mediators will degranulate. The release of histamine and other mediators will produce edema. edema को हम व्हील कहते हैं, and erythema यानी कि फ्लेयर. ठीक है, within a minute. This acute inflammatory reactions reaches its maximum intensity in 30 minutes and then disappear within next few hours. The disadvantage of skin testing is that it is inexpensive and simple to perform and allow the screening of several allergens. अब इसके अलावा पहला तो ये टेस्ट हो गया इंट्राडर्मल टेस्ट, इंट्राडर्मल स्किन टेस्ट हो गया. दूसरा होता है पैसिव क्यूटेनियस एनाफाइलैक्सिस, PCA. PCA टेस्ट इज अनदर इन विवो मेथड दैट इज यूज्ड टू डिटेक्ट IgE एंटीबॉडी इन द सीरम ऑफ द हाइपरसेंसिटिव एनिमल. इन दिस टेस्ट सेवरल डाइल्यूशंस ऑफ सीरम, इसमें वापस पढ़ते हैं. पैसिव क्यूटेनियस एनाफाइलैक्सिस test is another in vivo method that is used to detect IgE antibody in the serum of the hypersensitive animals. In this test, several dilution of serums from affected animals are injected into the skin of normal animal, and the allergen is inoculated intravenously after 24 to 48 hours. If positive reaction, acute inflammatory response develops at the injection site. Sometimes skin reactions are too mild to detect clearly. In such cases, even blue dye is injected intravenously into the test animal, which bind to albumin. Due to increased permeability at the site, dye labeled albumin comes out in the tissue fluid and produces a striking blue patches. The size of patch is proportional to the intensity of the inflammatory reaction. In vivo method of assessing type first hypersensitivity involves determination of total serum IgE antibody by radioimmunosorbent test, यानी कि RIST, radioimmunosorbent test. So in vitro method of assessing type first hypersensitivity involves determination of total serum IgE antibody by radioimmunosorbent test. तो ये तीसरा टेस्ट है. The assay is highly sensitive and can detect nanogram level of total IgE. A similar test called radioallergosorbent test, यानी कि RAST. तो ये चौथा टेस्ट है, ये है radioallergosorbent test. Allows detection of serum IgE specific to a given allergen. In this technique, allergen bound to beads are allowed to react with the test serum. The concentration of specific IgE bound to solid phase allergen is then measured by adding I-125 labeled anti-IgE and counting the bound radioactivity. Enzyme-linked immunosorbent assay ELISA has also been used to measure specific IgE antibody in atopic dogs. However, total serum IgE level does not always correlate well with the clinical severity. For example, a heavily pasteurized dog may reveal elevated level of IgE antibody. Therefore, assessment of allergen-specific IgE is more advantageous. तो ये था अभी तक टाइप फर्स्ट हाइपरसेंसिटिविटी. अब पढ़ेंगे हम टाइप सेकेंड हाइपरसेंसिटिविटी. Type 2 hypersensitivity. Antibody binding to cell surface antigen causes destruction of cell through a mechanism called type 2 hypersensitivity. This type of reaction is best exemplified by incompatible blood transfusion, where antibody is present in the recipient serum destroyed donor's red blood cells. Certain glycolipids are integral part of the erythrocyte plasma membrane and acts as antigen. They are water soluble. Physiological function of most of these antigens are not known except for the ABO system of humans. Which acts as a glucose transporter protein. These antigens found on the surface of erythrocytes are known as blood group antigen. Erythrocytes of man and animals are provided with different blood group antigen. They vary among different species with regards to their structure, antigenicity and complexity. Some blood groups antigens such as B system in the cattle and A antigen in the cat are most important from clinical point of view and are widely distributed in the population. Blood group antigen may be simple यानी कि J antigen in the cattle or may be complex, that is B antigen in cattle. These antigens are not unique to erythrocytes. They have also been identified in the several secretions, saliva and body fluid, and are positively absorbed into the erythrocyte. Natural antibodies यानी कि iso antibodies to red cells are formed in the man and animals without any previous exposure to the foreign blood group antigen via blood transfusion or another means. Iso antibody is usually of IgM type. Experimental evidences suggest that many of blood groups antigenic epitopes are shared by bacteria and plant cells. Hence, exposure to these antigens occur during the neonatal period that leads to antibody formation. Iso antibody formation is commonly seen in the humans ABO blood group system. अब इसका मैकेनिज्म क्या है टाइप 2 हाइपरसेंसिटिविटी का, वो पढ़ते हैं. Antibody mediated destruction of cells is the basic mechanism involved in type 2 hypersensitivity reaction. Example include incompatible blood transfusion, hemolytic disease of the newborn and cellular dysfunction. ब्लड ट्रांसफ्यूजन भी एग्जाम्पल है, हेमोलाइटिक डिसीज ऑफ न्यूबॉर्न भी एक एग्जाम्पल है, एंड सेल्यूलर डिसफंक्शन भी एक एग्जाम्पल है. Antibodies can mediate destruction of cell in 3 ways. First, the combination of antibody with the cell surface antigen activates the full complement system, generating membrane attack complex and formation of pores in the foreign cells. Second, antibody can mediate killing of cell by antibody dependent cell cytotoxicity mechanism. Third, a number of cells including NK cells, macrophages, monocytes, neutrophils and basophils express receptors for Fc region of antibody on their surface. These effector cells bind to the target cell through Fc receptor and initiate lysis of antibody coated target cells. Cytotoxicity mechanism of these cells include release of lytic components, tumor necrotic factor and perforin etc. Third, antibody bound to the foreign red cell antigen acts as opsonin, enabling phagocytic cells with Fc and C3b receptor to bind and phagocytose the antibody coated cells. In incompatible blood transfusion, rapid destruction of large number of erythrocytes results from type 2 reaction. In severe reaction, there is massive hemolysis and complement activation. This results into release of large amount of hemoglobin in the blood and urine. Lysed red blood cell are also activate blood clotting system, which leads to disseminated intravascular coagulation. Activated complement causes formation of anaphylatoxin and release of vasoactive agent due to mast cell degranulation. This substance provoke a vicious cycle where animals shows circulatory hypotension, bradycardia, and apnea. The affected animals exhibit sweating, salivation, lacrimation, diarrhea, and vomiting. Blood transfusion should be immediately stopped, and diuretic therapy may be resorted to maintain urine flow and to prevent hemoglobin-induced tubular necrosis. तो अब देखते हैं ब्लड ग्रुपिंग है तो ब्लड ग्रुप्स एंड हेमोलिटिक डिसीजेज इन डॉग में क्या होता है 11 इंडिपेंडेंट ब्लड ग्रुप सिस्टम होते हैं ए बी सी डी एफ जे के एल एम एन एंड टी आर हैव बीन आइडेंटिफाइड इन द डॉग ये 11 है मोस्ट ऑफ दिस एंटीजन आर इनहेरिटेड एज सिंपल मेंडेलियन डोमिनेंट ट्रेट. Blood typing in the dog is carried out by agglutination, hemolysis, and antiglobulin test. Before transfusion, blood typing is generally required in the dog because many dogs contain natural antibodies. Out of 11 blood group system, A system is highly immunogenic and is of clinical significance. About 2/3 of dogs have A blood group system. Only a minority of dogs shows presence of natural antibody to A antigen in A negative dogs. Spontaneous hemolytic disease in the newborn pups have been rarely reported. तो अब केटल की बात करें तो केटल में ये इसको अपन आगे पढ़ेंगे. So next in cattle blood group systems is very complex and so for 11 blood groups A, B, C, F, J, L, M, R, S, Z and T have been identified. Of this B and J group are most important. B or J, BJ. The B blood group on the cattle was first described in 1950 and has been extensively studied since then. At least 600 alleles have been identified in this group. The polymorphic characters of its alleles has been widely used by breed registries for percentage confirmation and parentage confirmation and for the maintaining identification of animal. This antigen has inherited in a combination known as phenogroups. In C blood group, there are 10 antigens which in combination form 90 phenogroups. The J antigen is actually a soluble lipid that is absorbed on the surface of the erythrocyte. It may be found in high concentration or in extremely low concentration. The J negative cattle possesses anti-J antibodies, concentration of which varies with seasonal variation. Transfusion of blood from J positive cattle to J negative cattle may cause transfusion reaction. However, transfusion reaction in bovine is of minor consequences. Hemolytic disease of newborn calves is rarely encountered, but many occur but may occur as a result of vaccination of cattle against anaplasmosis or babesiosis. Both the vaccines contain blood components as these are prepared from blood of cattle infected with anaplasma or babesia. While vaccine confer immunity in recipient animal against this protozoal disease, it produces antibodies against blood group antigen present in the vaccine. This is of little consequence to the animal itself. However, it often produces severe disease in suckling calves. Such vaccinated calf, when mated with bull carrying same blood antigen, eventually transfer iso antibodies against red blood cell antigen to the newborn calf via colostrum, causing hemolytic disease. A and F systems have been frequently implicated in HDNB in calves. The severity of HDNB in calves depends on amount of colostrum ingested. Calves are usually born healthy but soon show symptoms of respiratory difficulty and hemoglobinuria as a result of erythrolysis. In the acute case, death may occur after 24 hours. In less severe cases, the corpse develops anemia and jaundice and may succumb during the first weeks of life. Postmortem changes consist of splenomegaly, pulmonary edema, and dark kidney. Affected coughs shows antibodies on their red cells in the antiglobulin test. Death is due to massive erythrolysis and eventual DIC, disseminated intravascular coagulation. हॉर्स की बात करें तो इन द हॉर्स इवन इंटरनेशनली एक्सेप्टेड ब्लड ग्रुप सिस्टम इज ACDKPQ एंड U, यानी 1 2 3 4 5 6 7 7 हैं, हैज़ बीन आइडेंटिफाइड सो फॉर इन एडिशन टू फ्यू अदर विदाउट ऑफिशियल रिकॉग्निशन. As with other species, simple blood transfusion is of little significance in the equines. Hemolytic disease of newborn, however, is relatively common in as compared with other species. In the mule foal, जो म्यूल होता है फॉल बच्चा उसका, the condition has been reported more frequently, that is 10%, than in the horse foal, that is up to 2%. Information on blood group in equine is helpful in predicting where hemolysis will occur in the foal. Foal born out of mare sensitized by incompatible blood transfusion may suffer from hemolytic disease. But most commonly the syndrome is initiated when fetal red blood cells expressed antigen derived from stallion, whose blood group differ from the mare's. The pregnant mare divide of these antigens produce isoantibodies against them. The mechanism how fetal erythrocyte antigen reaches to the mare is not precisely known, but it is assumed that fetal erythrocytes gain access to the maternal circulation as a result of transplacental hemorrhages. During last trimester of pregnancy and during parturition, the possibility of leakage of erythrocytes is enormous. The first pregnancy is usually without any adverse effect in the foal. However, repeated pregnancies from the same stallion or other with blood group will cause further sensitization of them and predisposes the newborn foal to isoerythrolysis. Antibodies produced by the sensitized mare do not cross placenta to reach the foal. Foal receive them through the colostrum. Antibodies are absorbed by gastrointestinal mucosa and reach into the blood circulation. A type 2 hypersensitivity reaction ensues with resultant erythrolysis. Hence, foal is born healthy and becomes sick only after ingestion of colostrum. Clinical sign depends on amount of antibodies absorbed by the foal and usually appears after 6 to 48 hours. It includes anemia, depression, weakness, and slow and shallow respiration. Foals surviving beyond 48 hours show icterus of the mucous membrane and sclera. Hemoglobinuria, if present, it considered pathognomonic. Postmortem examination of the affected foals reveals anemia and icterus. The spleen and liver are enlarged. Characteristic clinical sign can readily diagnosed HDBN in foals. Laboratory confirmation is done by agglutination test using foals or stallions, erythrocyte, and mare's serum or colostrum. Hemolysis test is carried out by adding a fresh normal rabbit serum as a source of complement. Erythrocyte from the affected foal spontaneously agglutinate in vitro are Coombs positive. As the disease is common in the horse, mare with a history of neonatal erythrolysis should be tested to adopt preventive measures. Pregnant mare serum is tested for antibodies by direct antiglobulin test. A rising antibody titer indicate in utero sensitization. Newborn foal from such mare should not be allowed suckle colostrum and given frozen iso antibody free colostrum. अब sheep goat में देखते हैं. The blood group system in sheep goat resembles to that of cattle. 7 ब्लड ग्रुप्स इसमें मिलते हैं, A, B, C, D, M, R and X are recognized. Blood group B of sheep is also complex and is reported to have more than 50 different alleles. Ovine R system is analogous to bovine J system, है ना. ओवाइन का R सिस्टम और बोवाइन का J सिस्टम एनालॉगस है. In goats, 6 genetic system of erythrocyte antigen have been identified. इसमें यह है A, B, C, R, E and F. Like sheep, B system is complex and contain at least 21 different alleles. The E system has only 2 alleles, but has 4 phenotypes due to codominance. Hemolytic disease of newborn has been rarely reported in sheep and goat. Hemolysis test is used for identification of erythrocyte antigen. अब पिग की बात करते हैं तो पिग में 15 ब्लड ग्रुप्स मिलते हैं. 15 ब्लड ग्रुप सिस्टम A to O have been identified in pig. Of this, A blood group is most important. 2 blood group system A and O require a gene S. A and O को क्या रिक्वायर्ड होता है, जीन S रिक्वायर्ड होता है. S यानी secretor for their expression. When the secretor gene is in homozygous recessive state, A and O antigen are not produced. These antigens are found in the serum and passively absorbed into the erythrocyte after birth. A negative pig harboring anti-A antibody may show transfusion reaction after getting blood from A positive pig. Hemolytic disease of newborn piglet is an heterogenic disease which occurs as a result of vaccination of pig with crystal violet inactivated hawk cholera vaccine. Since this old-fashioned vaccine is prepared from pooled blood of viremic pig infected with hawk cholera, जो वायरेमिक पिग होते हैं ना, जो इन्फेक्टेड हॉक कोलेरा से होते हैं, वो. इट कंटेन्स एरिथ्रोसाइट एंटीजन. Sensitization with the vaccine also leads to isoantibody production, which in turn causes hemolytic disease in the pregnant. The affected piglets shows progressive weakness and ecteric mucous membrane. Surviving piglets may show jaundice and hemoglobinuria. Along with the hemolytic anemia, antibodies against platelets cause thrombocytopenia. अब केट्स की बात करें, केट में क्या होता है. AB is only the major blood group system identified in the cat. Population of cat does have 3 blood antigen A, B and AB. A अलग है, B अलग है और AB एक साथ है. A blood group is dominant and distributed in 75 to 95% of cat population. B blood group is prevalent in 5 to 25% of cat and AB is less than 1% of cat. This distribution, however, differs in different geographical region and pure breed cat breeds. Severe transfusion reaction have been reported, especially in B cats, because majority of B cats possesses anti-A of IgM class. IgM होता है ना एंटीबॉडी, उस क्लास का anti-A. Even very small amount of blood transfusion from A cat to B cat causes fatal shock. The affected cat shows hypotension, apnea and atrioventricular blockage. Atrioventricular block within few minutes of transfusion. Hemolytic disease of kittens has been reported rarely in Persian or Himalayan breeds. Kitten born from B positive queen mated with A positive sire suffers from the hemolytic disease. Affected kittens are anemic as a result of intravascular hemolysis. Hemoglobinuria may be noticed after several hours. Ecteric mucous membrane and splenomegaly are observed at postmortem. The queen serum contain antibodies to kitten or sire's erythrocytes. The blood typing in cat is carried out by agglutination and hemolysis test. नेक्स्ट है hemolytic disease of newborn in humans. अब humans की बात कर लेते हैं. In humans, HDNB occurs as a result of crossing of maternal IgG antibodies to fetal erythrocyte antigen to the fetus via placenta, causing erythrocyte destruction. Severe hemolytic disease, also called as erythroblastosis fetalis, is caused by rhesus incompatibility. It develops in Rh negative mother carrying Rh positive fetus. During pregnancy, the fetal erythrocytes are separated from the maternal circulation by a layer called trophoblast. At the time of delivery, however, fetal red cells gain access to the maternal circulation due to placental separation from the uterine wall. Rh positive fetal erythrocytes stimulate Rh positive specific B cell, which transform into plasma cells and memory cells. Plasma cells secrete IgM antibody, which kills fetal cells, and memory cells remain in the body. Subsequent pregnancy with Rh positive fetus results in activation of memory cells and formation of anti-Rh negative IgG antibodies, which cross the placenta and damage the fetal erythrocytes. Mild to severe anemia develops, which is overlooked. Many may become fatal. The breakdown product of hemoglobin, the bilirubin, which is lipid soluble, may accumulate in the brain and cause damage there. Hemolytic disease of the newborn can be prevented by the administration of antibodies to Rh antigen within 24 to 48 hours of delivery. इसके अलावा ब्लड ट्रांसफ्यूजन रिएक्शंस भी जो होती हैं वो भी हाइपरसेंसिटिव रिएक्शन है क्लास सेकेंड. Blood transfusion reaction have been reported in the dog, cat, horses, cows, pig and goat, and naturally occurring iso antibodies to most of the erythrocytes antigens are absent in the animal. First transfusion does not causes any discomfort or clinical reaction, hence cross matching or blood typing before first transfusion is not required. However, if the donor erythrocyte antigen is not identical to that of the recipient, it does stimulate antibody formation. The second blood transfusion from the same animal or the other animal with identical antigen would induce type 2 reaction mediated by circulating iso antibodies, which are usually of IgG type. There is rapid agglutination of erythrocyte and intra and extravascular erythrocyte destruction. The severity of transfusion reaction depends upon immunogenicity of the blood group antigen involved. The animal may suffer generalized anaphylactic shock, hemoglobinemia, and disseminated intravascular coagulation. Incompatible blood transfusion involves minor antigen have a long course. Incompatible blood transfusion involves minor antigen have a long course 4 to 14 days and cause progressive anemia and icterus. Direct antiglobulin test can be used for diagnosis. Cross matching using donor's red blood cell and recipient serums will result in positive slide agglutination test. There are some practical tips to avoid occurrence of acute blood transfusion reaction. For example, transfusion of A positive red cell in the dog, cat and horses, T positive red cells in the cattle, and R positive red cells in the sheep should be avoided. These blood groups antigens provoke a strong isoimmune response in the animal, thus cause severe reaction following subsequent exposure. Weak blood group antigens such as B, D, E in G in dog elicit formation of low titer iso antibodies and thus are little clinical significance. However, such sensitized animal may give positive antiglobulin test. Blood group and transfusion reaction in the man. 4 blood group phenotypes A, B and O are found in the human. Beside other such as RH, KID, KALE and Duffy, मतलब ये 4 फिनोटाइप्स तो मिलते ही हैं. इसके अलावा अदर जैसे RH भी है, किड है, केल है और डफ़ी है, इन लोगों ने भी अपने अपने कुछ फिनोटाइप्स दिए हैं. Incompatible blood transfusion such as infusion of type A blood into the type B recipient results in type 2 hypersensitivity reaction. Anti-A antibody presents in the recipient blood recognized as A antigen or transfused erythrocytes as foreign and initiate complement mediated erythrocyte. The clinical manifestation may have immediate or delayed onset. ABO incompatibilities are most commonly associated with immediate reaction in which complement mediated lysis is triggered by IgM antibodies. Hemoglobinemia and hemoglobinuria appears within 1 to 2 hours of transfusion. Patients show respiratory distress, disorientation, and cardiovascular collapse that may be fatal. Delayed transfusion reaction occur as a result of blood group antigen incompatibilities other than ABO incompatibilities. The patient receiving repeated transfusion of ABO compatible blood group produce antibodies against other antigen such as RH, Kidda, Kelly, and Duffy. The antibody of the IgG type, which is less efficient than IgM in activating complement mediated lysis. Hence many of donor red cells are destroyed extracellularly by agglutination, opsonization, and subsequent phagocytosis by macrophages. The reaction develops within 2 to 6 days of transfusion, reflecting secondary nature of responses. Symptoms symptoms are fever, low hemoglobin content, anemia, and mild jaundice. There may not be hemoglobinuria, as most of erythrocytes are killed at extracellular site. Now next is a type second hypersensitivity to drugs. तो drugs से भी hypersensitivity होती है. Certain drugs are capable of including type second reaction and cause hemolytic anemia. There are 3 mechanisms by which drug destroy erythrocyte. First है drugs such as penicillin, है ना, phenacetin and quinine etc. are passively absorbed onto the surface of the erythrocyte and render them immunologically foreign. In horses, penicillin induces hemolytic anemia has been reported. Antibodies to penicillin and penicillin-coated red cells can be demonstrated in the affected animals. A few drugs such as sulfonamide and phenylbutazone can cause agranulocytosis. ठीक है, agranulocytosis भी करते हैं. Similarly, drug induced thrombocytopenia is also reported. Second, drug and antibodies bind together and active complement, which in turn causes hemolysis of nearby cells. Thus, erythrocytes are killed as innocent by standards. Third mechanism involves the drug-induced modification of the cell membrane, allowing antibodies to be absorbed on the red cell for subsequent opsonization by phagocytes. Now next is the type second hypersensitivity in the infectious diseases. During infection, bacterial lipopolysaccharides, viruses of equine infectious anemia, and eluting disease, anaplasma, trypanosomas, and babesia may alter erythrocyte, making them immunologically foreign. Such erythrocytes are then eliminated by antibody and complement mediated lysis or phagocytosis by mononuclear cells. Now next is the type third hypersensitivity. Immune complex disease occur in the animal and man due to antigen-antibody complex included अभी हम पढ़ने जा रहे हैं टाइप थर्ड हाइपरसेंसिटिविटी. ठीक है, टाइप सेकंड खत्म हो चुका है, टाइप थर्ड समझते हैं. Immune complex diseases occur in the animal and man due to antigen-antibody complex induced pathological alteration including inflammation. Human complexes are formed immune complexes are formed normally and are removed by phagocytic cells from the system, but occasionally the formation of antigen and antibody complex are enhanced, which may get destroyed in tissue and leads to type 3 hypersensitivity disease. Broadly, the disease of immune complex origin can be classified into 3 groups. First है the persistent infection, where low grade persistent infection, that is the antigen, is present in the body and combined with the antibody produced against them lead to immune complex formation and damage in the tissue whether it gets deposited, जैसे कि viral hepatitis. Second is certain autoimmune disease also leads to immune complex mediated inflammation. However, it requires a continuous production of autoantibodies against self-antigen. The immune complexes thus formed are deposited in the various tissue leading to pathological implications. That is the systemic lupus erythematosus. तीसरा है the formation of immune complexes in the tissue due to repeated exposure of antigen and formation of antibodies. The antibodies produced are of IgG class, that is the farmer's lungs, FARMER, farmer's lungs. Farmer's lungs एक example है जिसमें IgG antibodies बनती हैं. Due to inhalation of mold and fungal spores, local immune complexes are formed in the lung alveoli, leading to alveolitis and pneumonia. Immune complexes are formed at 2 places. ऐसे 2 जगह बनते हैं. In blood circulation and streams and circulate to lodge in the vascular channel such as the capillaries, renal, glomerulus and choroid plexus and or synovial membrane. और दूसरा है कि टिश्यू में बनते हैं. पहला तो ब्लड सर्कुलेशन में बनते हैं, दूसरा टिश्यू में बनते हैं. इन द टिश्यू विच रिमेन्स लोकलाइज्ड एट साइट ऑफ इम्यून कॉम्प्लेक्स फॉर्मेशन. The size and character of antibody are more important than the nature of antigen. A small size immune complex remain in the circulation and are not trapped in the blood vessel or capillaries, while large size immune complexes are soluble and are quickly removed by phagocytosis. However, the medium size immune complexes remain in the circulation for a longer period. They can fix the complement and are trapped in the small capillaries and renal glomeruli, leading to inflammation. Response immune complexes may induce a variety of inflammatory lesions. The binding of antibody to antigen also interact with the complement system, leading to generation of C3A and C5A, which are having an anaphylactic and chemotactic property and cause the release of vasoactive amines from the mast cell and basophils. These chemicals, mediators of inflammation, act on the blood vessel and cause increase in the vascular permeability, attract phagocytic cells. Immune complex are also interact with Fc receptor of platelets, leading to their aggregation and microthrombus formation, which further facilitate an increase in vascular permeability through the action of vasoactive amines. The phagocytic cell present at the site of immune complex deposition may attempt to phagocytose the complexes and are unable to do so because of their trapping in tissue. Thus, these phagocytic cells release lysosomal enzyme at the site of immune complex deposition, leading to further tissue damage. The class of immunoglobulins. The class of immunoglobulin can influence the deposition of immune complexes. In experiments with SLE model in mice, it has been observed that IgM and IgG2A participate in the immune complex formation. However, once complexes are deposited in the tissue, complement makes them soluble again. The solubilization appears to occur by insertion of complement C3b and C3d fragments into the complexes. The complexes are continuously being deposited in the normal individual and are removed by solubilization process. But in case of complementemic complement system कम हो जाए, the solubilization process is either absent or decreased, leading to prolonged deposition of immune complexes. The solubilization defect have been observed in the sera from patients with systemic immune complex diseases. The experimental model of all these three classical type of immune complexes mediated disease are, तो इसमें हम पहला वाला पढ़ते हैं। पहला वाला है सीरम सिकनेस। तो फर्स्ट है सीरम सिकनेस। इन सीरम सिकनेस सर्कुलेटिंग इम्यून कॉम्प्लेक्सेस डिपॉजिटेड इन द टिश्यू initiate inflammatory process leading to glomerulonephritis and arthritis. It has been observed as a complication of serum therapy. For example, in the diphtheria, anti-diphtheria horse serum is given in the man, which is recognized as foreign protein, and the body initiates an immune response against horse serum. Thus, the immune complexes are formed, leading to serum sickness. Serum sickness can also be produced in the rabbit by administering intravenous injection of foreign soluble proteins such as BSA. After 1 week, antibodies are formed, which either be circulating or bind with an antigen to make immune complexes. These immune complexes are removed by mononuclear phagocytic system slowly and persist in the circulation. Formation of immune complexes leads to fall in total complement level in the blood. The clinical sign of serum sickness can be observed as an antigen antibody and C3 deposits on glomerular basement membrane and in small vessels. As the complexes are removed from there either by phagocytosis or by solubilization by complement, animal recover and disease may take a chronic course by continued daily administration of antigen. Second है autoimmune complex disease. In systemic lupus erythematosus, a range of autoantibodies can be detected such as anti-RBC, anti-nuclear, anti-DNA, etc. The deposition of immune complexes are observed in glomeruli and choroid plexus. तो इसको कहते हैं autoimmune complex disease. Next है Arthus reaction. Arthus reaction takes place at local site in and around the wall of small blood vessels and can be demonstrated in skin. It can be observed in the animal by administration of repeated injection of antigen until an appreciable amount of precipitating IgG antibody appears. Then on subcutaneous or intradermal injection of antigen gives inflammatory reaction within 4 to 10 hours of administration. There is marked edema and hemorrhages at the site of injection which disappears after 48 hours. Antigen antibody complement can be demonstrated around the wall of a small blood vessel in the lesion, which is also infiltrated by neutrophils followed by mononuclear cells. There is intravascular clumping of platelets leading to occlusion of blood vessel and necrosis. Now disease associated with the immune complexes. तो अब जैसे इसमें हाइपरसेंसिटिविटी थर्ड हम पढ़ रहे हैं जिसमें इम्यून कॉम्प्लेक्सेस मीडिएटेड होता है ये, तो इससे कौन कौन सी डिसीज़ होती हैं वो पढ़ लेते हैं। जैसे ग्लोमेरुलोनेफ्राइटिस हो गया, तो इम्यून कॉम्प्लेक्सेस मीडिएटेड ग्लोमेरुलोनेफ्राइटिस वाज कंसिडर्ड एज रियर डिसीज entity till 1970, but now with the advancement of the immunopathology, it accounts substantial morbidity and mortality, not only in the animal but also in the man. In immune complexes mediated glomerulonephritis, the antigen antibody complexes are deposited in the glomerulus, leading to alteration in structure and function. It has been reported in the middle-aged dog, especially associated with the chronic skin lesions, neoplasia and hyperadenocorticism. In animals, the immune complexes mediated glomerulonephritis has been found to occur in various other disease condition listed below. तो इसमें कुछ लिस्ट दी है एनिमल्स कौन कौन से हैं, जैसे बोवाइन में बोवाइन वायरल डायरिया, फिर ओवाइन में कैम्पाइलोबैक्टीरियोसिस, पोर्साइन में स्वाइन फीवर है ना, इक्वाइंस में इक्वाइन इन्फेक्शियस एनीमिया एंड स्ट्रेंगल, और इसके बाद कैनाइन में ब्रूसलोसिस, फिर लिम्फोसार्कोमा, फिर मास्टोसाइटोमा, एडिनो वायरस इन्फेक्शन, इसके बाद पायोमेट्रा, फिर सिस्टेमिक ल्यूपस एरिथ्रोमेटोसिस। अब इसकी पैथोजेनेसिस क्या है, ये जो हम पढ़ रहे हैं इस समय ग्लोमेरुलोनेफ्राइटिस, इसकी पैथोजेनेसिस क्या है? इसकी पैथोजेनेसिस है इम्यून कॉम्प्लेक्सेस आर फॉर्म्ड आइदर थ्रू endogenous or exogenous antigen determinant in the host tissue. The antigen implanted in the host tissue is recognized by the circulating antibodies, which binds with the antigen, and the process is known as in situ immune complex formation. This type of immune complex mediated glomerulonephritis has been observed in the most cases of men and animals. In another type, the immune complexes are formed in circulation and they get deposited in the wall of medium or small sized blood vessel, such as the glomerular capillaries. Such complexes require high level of antigen as seen in certain persistent viral infection. Immune complexes are deposited within glomerulus either of the four anatomical location. पहला कहाँ हो सकते हैं ये? सब epithelial beneath the visceral epithelial cells. दूसरा सब endothelial हो सकते हैं beneath the endothelial cells. Intramembranous हो सकते हैं in lamina densa of the basement membrane. और चौथा हो सकते हैं mesangial cells outside of the glomerulus basement membrane in the mesangial cells. तो अब इनके बारे में अपन पढ़ते हैं, चारों के बारे में. पहला है सब एपिथेलियल. अगर एपिथेलियल की बेसमेंट के नीचे मिलता है तो उसमें द इम्यून कॉम्प्लेक्सेस आर डिपॉजिटेड इन सब एपिथेलियल रीजन व्हेन दे आर कंपोज्ड ऑफ केटायनिक चार्ज एंटीजन और कॉम्प्लेक्स ऑफ लो एविडिटी. The immune complexes having high cationic charge may cross glomerular basement membrane and bind with the anionic charged epithelial cell cytoplasm. In immune complexes with low avidity, antibody dissociated at the inner part of the glomerular basement membrane. The antigen then may diffuse across the membrane and is trapped on epithelial cell cytoplasm and subsequently rebind to its antibody. Thus, subepithelial immune complexes may result from either in situ or formation of circulating antigen antibody complexes. दूसरा होता है subendothelial. Those immune complexes are deposited in subendothelial area which are having natural or anionic charged antigen and thus cannot penetrate the barrier of glomerular basement membrane. Antibodies having high avidity may also tend to localize in subendothelial area. The immune complexes localized in subendothelial area have been found to be associated with more intense inflammation. तीसरा कहाँ हो सकते हैं? इंट्रामेम्ब्रेनस हो सकते हैं. दिस इज़ वेरी रेयर पैथोलॉजिकल कंडीशन. इन इंट्रामेम्ब्रेनस डिपोजिशन इन ऑफ द इम्यून सिस्टम of immune complexes, there is an autoimmune humoral response against glomerular basement membrane in which the indigenous structure glomerular antigen acts as autoantigen. और चौथा हो सकता है जो मेसेंजियम है, मेसेंजियम है ना, मेसेंजियम, क्योंकि मेसेंजियल सेल्स होती है ना, जो मेसेंजियल सेल्स, तो मेसेंजियम में the mesengium is composed of specialized cells and extracellular matrix and is consistently diffused by macromolecules such as infectious agent or immune complexes. Immune complexes accumulates in matrix after ultrafiltration and subsequently localized in mesangial cells through endocytosis. It results in proliferation of mesangial cells and production of excess mesangial matrix. अब इसका clinical finding क्या है? Immune complexes mediated glomerulonephritis has been well studied in canines. Initially, there is no clinical symptoms even after the disease started with glomerular injury, but is not sufficient to produce increased capillary permeability. However, one can detect the high protein level in the urine of patient as about 200 milligram of protein may pass through the urine per day. This leads to hypoproteinemia, edema, ascites, and hypercholesterolemia. The low level of albumin in the blood circulation may also result into hypovolemia. there are elevated level of serum cholesterol and triglycerides leading to hypolipidemia. In advanced cases, blood urea nitrogen level increases, resulting in azotemia and uremia characterized by anorexia, vomiting, diarrhea, melena, polydipsia, polyuria, dehydration, anemia, convulsions and weakness भी होता है. These signs occur with a functional loss of about 75% nephrons, reduced glomerular blood flow, and filtration that may lead to hypoxia, cell necrosis, interstitial inflammation, and fibrosis. Renal osteodystrophy can also be observed in patients with immune complex mediated nephritis because of increased renal retention of phosphorus, ineffective renal conversion of active form of vitamin D, which leads to hypocalcemia, increased production of parathermone, and eventually decalcification of bone. Now comes to lesions. ग्लोमेरुलोनेफ्राइटिस, सबसे पहले ग्लोमेरुलोनेफ्राइटिस ही होता है. ऑन नेक्रोप्सी, pale and firm kidney are seen with little fibrosis along the capsule, which is difficult to remove from kidney parenchyma. There is a loss of clear line of demarcation between cortex and medulla. क्योंकि फाइब्रोसिस हो जाती है ना, तो मेडुला कॉर्टेक्स बीच में कोई डिमार्केशन बचेगा नहीं. देयर इज हाई लेवल ऑफ प्रोटीन इन द यूरिन प्रेजेंट इन द यूरिनरी ब्लैडर. यूरिनरी ब्लैडर वाले में बहुत ज़्यादा प्रोटीन मिलेगा, हाई लेवल ऑफ प्रोटीन प्रेजेंट. माइक्रोस्कोपिकली 4 बेसिक टाइप ऑफ चेंजेस कैन बी ऑब्जर्व्ड इन द किडनी. पहला है प्रोलिफरेशन ऑफ मेनिंजियल सेल, थिकनिंग ऑफ बेसमेंट मेम्ब्रेन, इन्फिल्ट्रेशन ऑफ ल्यूकोसाइट्स इन इंजर्ड ग्लोमेरुलाई, हाइलाइनाइजेशन and sclerosis. Based on the lesion described above, the glomerulonephritis of immune complex origin is divided into 4 categories. तो ये glomerulonephritis को 4 भागों में बांटा है based on lesions. पहला है proliferative glomerulonephritis, दूसरा है membranous glomerulonephritis, तीसरा है membranoproliferative glomerulonephritis और चौथा है mesengiosclerotic glomerulonephritis. तो पहला है proliferative glomerulonephritis. This form of immune complex mediated glomerulonephritis is characterized by increase in number of cells within the glomerulus, such as epithelial cell, endothelial cells, and mesangial cells. Of which mesangial cells proliferation is most markedly seen. It is also known as mesengioproliferative इन मेडिकल साइंस में इसको हम मेसेंजियो प्रोलिफरेटिव कहते हैं. ठीक है. इन दिस टाइप ऑफ ग्लोमेरुलोनेफ्राइटिस एडहेशंस बिटवीन बॉमन्स कैप्सूल एंड ग्लोमेरुलर टफ्ट आर सीन एज सब एक्यूट और क्रोनिक लीजन इन द हॉर्सेस एंड रुमिनेंट्स. तो स्पेशली ये सब एक्यूट या फिर क्रोनिक लीजन मिलते हैं हॉर्स और रुमिनेंट्स में. इसमें ग्लोमेरुलाई और जो बॉमन्स कैप्सूल होता है वो आपस में स्टिफ हो जाता है या उसका एक टफ्ट बन जाता है. इम्यूनोफ्लोरेसेंस और इम्यूनोप्रोक्सीडेज स्टेनिंग टेक्निक्स रिवील्स द प्रेजेंस ऑफ इम्यून कॉम्प्लेक्सेस विथ IgG और IgM इन कैपिलरी वॉल एंड इन मेसेंजियम. मेम्ब्रेनस ग्लोमेरुलोनेफ्राइटिस. मेम्ब्रेनस ग्लोमेरुलोनेफ्राइटिस दूसरा है जिसमें इज कैरेक्टराइज्ड बाय मार्क्ड थिकनिंग ऑफ ग्लोमेरुलर बेसमेंट मेम्ब्रेन. इम्यूनोफ्लोरेसेंस और इम्यूनोप्रोक्सीडेज स्टेनिंग ऑफ किडनी सेक्शन में रिवील द प्रेजेंस ऑफ यूनिफॉर्मली कोर्स ग्रेनुलर डिपॉजिट. प्रेजेंस ऑफ यूनिफॉर्मली कोर्स ग्रेनुलर डिपॉजिट्स ऑफ होस्ट इम्यूनोग्लोबुलिन IgG और कंप्लीमेंट यानी कि C3 within the wall of glomerular capillaries. Chronic membranous glomerulonephritis is commonly seen in cats and dogs. Next है membranoproliferative glomerulonephritis. This is a composite form of proliferative and membranous glomerulonephritis. मतलब ये दोनों ऊपर हमने जो दो पढ़े हैं उन दोनों का मिला जुला है ये. Is in which both type of lesions are recorded. In this form, glomerular tuft and capillary wall become thickened along with increased infiltration of leukocytes. Irregular granular accumulation of immune complexes can be demonstrated in wall of glomerular capillaries and within the expanded mesangial matrix. The type of glomerulonephritis is most commonly seen in dog. तो ये वाला जो है डॉग में commonly मिलता है membranoproliferative glomerulonephritis. Next है mesangiosclerotic glomerulonephritis. It can be observed due to prolonged glomerular injury which occurs after the either of three types described above. In this form, glomerular tufts are extensively hyalinated. Hyaline बन जाता है, hyaline होता है ना, hyalinated, which becomes avascular and adhere to Bowman's capsule. This is complete destruction of glomerular cytoarchitecture and immune complexes can be seen in capillary wall and within the mesangium. Besides the pathological lesion in intraglomerular space, there is a significant tubulointerstitial lesion on chronic glomerulonephritis. The type and extent of lesion depends on quality and quantity of immune complexes and their site of deposition. However, there is some lesions overlapping to each other needs confirmation through IFT or immunoperoxidase staining. तो ये 4 टाइप के होते हैं ग्लोमेरुलोनेफ्राइटिस. अब हम आगे पढ़ते हैं. नेक्स्ट होता है rheumatoid arthritis. Rheumatoid arthritis is a bilateral erosions polyarthritis caused by deposition of immune complex within the synovial tissue. This type of arthritis occurs in men and dogs. Rheumatoid arthritis is an intra-articular deposition of immune complexes due to production of autoantibodies against self-immunoglobulin यानी कि self antigen. Thus, the altered immunoglobulin of host act as antigen and antibody to this immunoglobulin is known as rheumatoid factor RF, which is an autoantibody. तो जो अल्टर्ड इम्यूनोग्लोबुलिन्स होते हैं उन्हीं के अगेंस्ट वापस से इम्यूनोग्लोबुलिन बन जाते हैं और ये इम्यूनोग्लोबुलिन एक्चुअल में आरएच फैक्टर कहलाते हैं या रिमोटॉइड फैक्टर कहलाते हैं. तो इसको ऑटो एंटीबॉडी कहते हैं. द ऑटो एंटीबॉडी RF बाइंड विथ FC पोर्शन ऑफ होस्ट इम्यूनोग्लोबुलिन जिसको बेसिकली वो जो कि एंटीजन होता है एंड दस फॉर्म्ड कॉम्प्लेक्स that are deposited in the synovium leading to complement activation, chemotaxis of inflammatory cell and proliferation of synovial cells. There is configurational changes in the FC portion of host IgG after its binding to an unknown antigen. This change in the immunoglobulin render it foreign to the host and initiates the production of autoantibodies. It may also happen through a breach of self-tolerance owing to abnormalities of the immunoregulation. In some cases of the men with rheumatoid arthritis, decreased mitogenic responsiveness of lymphocytes leading to energy. In some cases of men with rheumatoid arthritis, decreased mitogenic responsiveness of lymphocyte. जो lymphocyte का माइटोसिस होता है ना, माइटोजेनिक रिस्पॉन्स नहीं हो पाता और डिक्रीज हो जाता है, तो माइटोसिस होता नहीं है. तो इसके कारण लीडिंग टू एनर्जी. तो लीडिंग टू एनर्जी एक स्टेट होती है, इसको पढ़ लेना अलग से. वंस द डिफेक्ट इन द होस्ट इम्यूनोग्लोबुलिन immunoregulation is established, the plasma cell activated B cell within the synovium and the regional lymph nodes produces large amount of autoantibodies. These autoantibodies form immune complexes with host immunoglobulin and deposit in synovium leading to inflammation and pannus formation, PANNUS, pannus formation. The inflammation in the synovial membrane occurs due to fixation of complement and immune complexes and release of C3 and C5a. The pannus is intra-articular formation of granulation tissue which arises from inflamed synovium. है ना, साइनोवियम के इन्फ्लेम्ड हो जाने से पेनस फॉर्मेशन होता है और पेनस फॉर्मेशन बेसिकली इंट्रा आर्टिकुलर फॉर्मेशन ऑफ ग्रेनुलेशन टिश्यू है. तो एंड एक्सटेंड अक्रॉस द आर्टिकुलर कार्टिलेज, जो कि पूरी आर्टिकुलर कार्टिलेज में फैलता है. The inflammatory cell includes proliferating synoviocytes, and fibrinolectin are main component of the penis. Clinically, the rheumatoid arthritis is characterized by bilaterally symmetric progressive polyarthritis, most commonly affecting carpal, metacarpal, tarsal, metatarsal joints. The other joints which are involved in the rheumatoid arthritis includes interphalangeal, interphalangeal, stifle, elbow, and intervertebral joints. The patient suffer from joint pain, stiff gait, and periarticular swelling, along with fever, depression, and anorexia. On clinical examination, such joints are found to be swollen and painful. Diagnosis can be made by demonstration of rheumatoid factor in the serum of the patient. Immune complex mediated polyarthritis commonly occur in men, dog, and cat, and in other animals. It has not been documented so far. For treatment of immune complex mediated disorder in the man and animal, immunosuppressive and anti-inflammatory drugs are given. Treatment with corticosteroid has been found to have a recovery of about 90% in arthritis patient. However, it may have its other side effect also, which should be looked into before start of treatment. तो ये सब था अभी तक हमारा टाइप 3 हाइपरसेंसिटिविटी, जो कि इम्यून मीडिएटेड होती है. अब हम आते हैं नेक्स्ट टाइप 4 हाइपरसेंसिटिविटी पर. तो वी हैव लर्न दैट एंटीबॉडी मीडिएट्स फर्स्ट 3 टाइप ऑफ हाइपरसेंसिटिविटी रिएक्शन, टाइप 1, 2, and 3, which occur within few minutes to few hours of re-exposure to allergens. Another type of hypersensitivity also occur, which is known as type 4 hypersensitivity and is mediated by sensitized T cells. When some antigen are injected into the skin of sensitized animal, acute inflammatory reaction takes several hours to develop and become clinically obvious between 24 to 72 hours. Hence, this type of hypersensitivity reaction is also called as delayed type of hypersensitivity, DTH. It results from interaction between the allergen, macrophages, and the T cells. The reaction is characterized by red, indurated, painful swelling in which macrophage is the principal component. इसमें indurated painful swelling होती है. ठीक है. The reaction is characterized by red, indurated, painful swelling in which macrophage is the principal component. Historically, Robert Koch first observed the phenomena about a century ago. 100 साल पहले ही रॉबर्ट कोच ने टाइप 4 हाइपरसेंसिटिविटी निकाल दी थी. He found that individuals infected with the Mycobacterium tuberculosis showed local inflammatory reaction when injected intradermally with mycobacterial culture filtrate. The reaction was named as the tuberculin reaction, which has now been considered the most classical example of DTH. Subsequently, it was found that a variety of bacterial and other agents could cause this phenomenon. As the term suggests, type 4 hypersensitivity is detrimental and at times causes extensive tissue damage, but in many instances, especially against intracellular pathogen and contact antigen, it plays important protective role. तो जैसा कि टर्म कहता है, ये बहुत ही डेट्रीमेंटल है टाइप 4 हाइपरसेंसिटिविटी, क्योंकि इसमें जितना ज़्यादा समय लगता है टिश्यू डैमेज उतना ज़्यादा होता है. वैसे भी ये इंट्रासेलुलर पैथोजेंस के कारण होने वाला हाइपरसेंसिटिविटी है, और कॉन्टैक्ट एलर्जी जो होती है, कॉन्टैक्ट एंटीजन के कारण होने वाली जो हाइपरसेंसिटिविटी है वो भी इसी कारण होती है. तो अब सबसे पहले हम रॉबर्ट कोच का ट्यूबरकुलिन रिएक्शन पढ़ेंगे. द ट्यूबरकुलिन इज द एंटीजेनिक एक्सट्रैक्ट डिराइव्ड फ्रॉम माइकोबैक्टीरियम बोविस, माइकोबैक्टीरियम ट्यूबरकुलोसिस एंड माइकोबैक्टीरियम एवियम. In past, several types of tuberculin were prepared as a reagent for the diagnosis of tuberculosis in the animal. But currently, purified protein derivatives यानी PPD of tuberculin is most commonly used. Normal healthy animal when injected with tuberculin does not show any reaction. On the other hand, when tuberculin is injected into an animal already infected with the tuberculosis bacilli or sensitized with mycobacterium antigen, an acute indurated swelling develops at the site of injection after 24 hours. The swelling reaches its maximum within 48 to 72 hours. The skin reaction may persist for several weeks before it disappears. तो 2 दिन से 3 दिन तक, वैसे तो 24 घंटे से चालू हो जाती है, लेकिन जो रिएक्शन है वो मैक्सिमम हो जाती है दूसरे दिन से तीसरे दिन में. द स्किन रिएक्शन में persist for several weeks before it disappears. If the animal is highly sensitized, a severe reaction considered बहुत वीक तक वो persist करती है, फिर बाद में डिसअपेयर होती है. ठीक है. इफ़ द एनिमल इज़ हाईली सेंसिटाइज्ड, ए सीवियर रिएक्शन कंसिस्टिंग ऑफ टिश्यू डिस्ट्रक्शन एंड नेक्रोसिस अकर इन द डर्मिस. तो टिश्यू डिस्ट्रक्शन भी हो जाता है, नेक्रोसिस भी हो जाती है डर्मिस की. हिस्टोलॉजिकली द रिएक्शन इज कैरेक्टराइज्ड बाय इंगॉजमेंट ऑफ ब्लड वेसल, edema and heavy infiltration with lymphocytes and macrophages. Presence of neutrophil is significant number may be seen in the early stage. तो शुरुआत में तो न्यूट्रोफिल आएंगे, पर बाद में वहाँ ब्लड वेसल्स का इंगॉजमेंट हो जाएगा, इडीमा होगा, हेवी इन्फिल्ट्रेशन होगा, और विथ द लिम्फोसाइट एंड मैक्रोफेजेस. Mechanism of DTH. It is evident from the classical tuberculin reaction that to occur DTH reaction as for other hypersensitivity reaction, in first step animal has to be sensitized. Following re-exposure with the same antigen, clinically obvious skin reaction occur only after 24 hours, which indicate effector function of the sensitized T cells. Thus, mechanism of DTH reaction can be explained in two phases. First phase is the sensitization and second phase is the effector phase. देखो, in the sensitization phase, the offending antigen causes activation of T helper cell in 1 to 2 weeks. The antigen is processed by antigenic presenting cells APC such as the Langerhans cells and macrophages, and is presented to helper cells subpopulation of T cells in association with MSC class second molecule. The antigen presenting cells are thought to pick up the antigen from the site of infection or injection and bring them to the regional lymph nodes where the T cells are activated and clonically expanded. Usually CD4 plus T cells are activated and participate in the DTH reaction. These cells are specially called DTS cells. In some cases, cytotoxic T cells are also activated. Activated T cells may be present at the site of infection और सर्कुलेट इन द ब्लड. तो ये था सेंसिटाइजेशन फेज़ हो गया, जिसमें ये फेज़ चलती है टी हेल्पर सेल की, जो कि 1 से 2 सप्ताह तक चलती है. इसमें जो लैंगरहैंस सेल्स हैं वो एपीसी हैं और वो मैक्रोफेज भी हो सकते हैं, और वो टी हेल्पर सेल की तरफ एमएससी क्लास सेकंड मॉलिक्यूल से इस एंटीजन को ब्लड सर्कुलेशन में लेके जाते हैं. जहाँ से भी एंटीजन पकड़ा है इन्होंने, वहाँ से ब्लड सर्कुलेशन की तरफ लेके जाते हैं. अब दूसरा फेज़ होता है इफेक्टर फेज़. द इफेक्टर फेज़ इन induced by subsequent exposure with the similar antigen. Following intradermal injection, a part of antigen is taken by the dendritic cell, which migrate to draining lymph node to activated sensitized T DTS cells. फिर से पढ़ता हूँ. द इफेक्टर फेज इज इंड्यूस्ड बाय सब्सिक्वेंट एक्सपोजर विद द सिमिलर एंटीजन. तो एक ही एंटीजन का बार बार एक्सपोजर होगा तो भी होगा. फॉलोइंग इंट्राडर्मल इंजेक्शन, a part of antigen is taken by a dendritic cell which migrate to the draining lymph node to activated sensitized TDTH. Antigen are also locally absorbed and some in contact with the circulating TDTH, which in turn adhere to endothelial cells and migrate to the antigen deposition site. Locally, antigen also attract neutrophil and monocytes. तो इफेक्टर फेज़ में क्या होता है कि पहले से ही जो सेंसिटाइजेशन वाली जो फेज़ थी, उसमें जो टी हेल्पर सेल्स हैं वो कन्वर्ट हो जाती हैं टी DTS सेल्स में, अब इफेक्टर सेल्स में क्या होता है कि ये सीधा जो TDTH रहती है वो एक्टिवेटेड हो जाती है, बस और कुछ नहीं है। Macrophage functions are the principal effector cells in the DTH reaction. The cytokines secreted by the activated TDTH cells causes increased expression of adhesion and macrophage second and MSC class second molecule. तो जो DTH रिएक्शन है उसमें मैक्रोफेज का सबसे ज़्यादा इम्पोर्टेंस होता है, क्योंकि उसमें जो साइटोकाइंस हैं वो DTH सेल्स से एक्सक्रीट होते रहते हैं और MSC क्लास सेकेंड मॉलिक्यूल का एक्सप्रेशन और एडिशन करवाते हैं। दिस रिजल्ट्स इन द माइग्रेशन ऑफ ब्लड मोनोसाइट्स इन द सराउंडिंग टिश्यू. They are differentiated into activated macrophage equipped with enhanced capacity of phagocytosis and destruction of the microorganisms. This activated macrophage are also express increased level of MSC class second and cell adhesion molecule so as to function more effectively as antigen processing cell. तो ये एंटीजन प्रेजेंटिंग सेल तो होती है, ये साथ में एंटीजन प्रोसेसिंग सेल्स भी होती हैं ये मैक्रोफेजेस. It has been observed in the cattle that by 24-hour injection site is mainly infiltrated with gamma lambda positive WC positive T cells along with few neutrophils. The reaction is apparently viable. The reaction is apparently visible by 24 hours and its intensity may reach peak level within 48 hours. Vasoactive substances are released by T cells and also from basophils and mast cells, which are attracted to the site in the response to cytokines. तो 24 आवर्स के अंदर जो T सेल्स हैं उसके अलावा न्यूट्रोफिल्स भी एक्टिवेटेड हो जाती हैं और कुछ वैसोएक्टिव सब्सटेंसेस रिलीज होते हैं बाय T सेल्स and also from basophils and mast cells, which are attracted to the site in the response to cytokines. More and more T cells are attracted to the site in the response to T cell derived molecules, यानी कि ऑटोक्राइन फंक्शन, तो खुद से ही खुद को एक्टिवेट करते हैं ये. Only about 5% of infiltrated T cells are antigen specific, यानी कि TDTH cells, and the rest are non-specific to the inducing antigen. At the peak of response, phenotypically lymphocytes are TCR alpha beta positive, CD4 positive and CD8 positive T cells. Proteases and reactive oxygen metabolite released from the activated macrophages cause tissue destruction. The area is swollen and indurated due to massive infiltration of macrophages and lymphocytes. Fibrin deposition and inflammatory edema ये भी हो जाता है. तो एरिया क्या होगा, स्वेलन हो जाएगा, इंड्यूरेटेड हो जाएगा ड्यू टू मैसिव इन्फिल्ट्रेशन ऑफ मैक्रोफेजेस, क्योंकि वहाँ मैक्रोफेजेस बहुत ज़्यादा आ जाते हैं एंड लिम्फोसाइट्स भी आ जाते हैं वहाँ पे. फाइब्रिन डिपोजिशन होता है और इन्फ्लेमेटरी एडिमा होता है. द इंजेक्टेड एंटीजेंस आर इवेंचुअली इंजेस्टेड एंड डिस्ट्रॉयड एंड द स्वेलिंग ग्रेजुअली डिसअपियर्स इन सेवरल वीक्स. तो एंटीजन धीरे धीरे इंजेस्टेड हो जाएगा, डिस्ट्रॉय हो जाएगा और स्वेलिंग धीरे धीरे ख़त्म हो जाएगी. इसका बहुत अच्छा फिगर इसमें दिया हुआ है. अब इसमें मीडिएटर्स कौन कौन से होते हैं. मीडिएटर्स ऑफ DTH रिस्पॉन्स. अ वेराइटी ऑफ साइटोकाइंस एंड कीमोकाइंस सिक्रेटेड बाय एक्टिवेटेड लिम्फोसाइट्स एंड मैक्रोफेजेस मीडिएट्स DTH रिस्पॉन्स. पैटर्न ऑफ साइटोकाइंस सिक्रेटेड suggests that TDTH cells are derived from T helper cell population. Gamma interferon यानी कि interferon gamma is the most important of all and activates macrophages and promote increased expression of class second molecule on them. तो इसमें देखने में आया है कि DTH में सबसे ज़्यादा इम्पॉर्टेंट सेल होती है T हेल्पर सेल, और गामा इंटरफेरॉन जो होता है इंटरफेरॉन गामा ये बहुत इम्पॉर्टेंट साइटोकाइन है जो कि MSC क्लास सेकेंड मॉलिक्यूल के एक्सप्रेशन को मैक्रोफेज के ऊपर बढ़ाता है. इंटरफेरॉन गामा एंड ट्यूमर नेक्रोसिस फैक्टर बीटा along with tumor necrotic factor alpha and interleukin 1 act on the endothelium of the neighboring blood vessel and facilitate extravasation of monocytes and other non-specific inflammatory cells. तो फिर से मैं पढ़ता हूँ कि जो इंटरफेरॉन गामा है, ट्यूमर नेक्रोटिक फैक्टर बीटा है और T1 नेक्रोटिक फैक्टर अल्फ़ा है और इंटरलेकिन 1 है। ये एंडोथेलियम ऑफ नाइबरिंग ब्लड वेसल पे काम करते हैं और वहाँ से एक्स्ट्रा विसेशन करवाते हैं ऑफ द मोनोसाइट एंड अदर नॉन स्पेसिफिक इन्फ्लेमेटरी सेल। मोनोसाइट जब बाहर निकलेगी तो मैक्रोफेज में कन्वर्ट हो जाएगी। द प्रोसेस इन्वॉल्व्स इंक्रीज्ड एक्सप्रेशन ऑफ एडेशन मॉलिक्यूल ऑन द एंडोथेलियम एंड इंक्रीज्ड वेस्कुलर परमेबिलिटी. Neutrophils appears early in the reaction and are maximum by 6 hours, after which number declines. इसके बाद धीरे धीरे नंबर घट जाते हैं। 6 घंटे तक न्यूट्रोफिल सबसे ज़्यादा एक्टिवेटेड रहते हैं, बाद में डिक्लाइन हो जाते हैं। मोनोसाइट स्टार्ट अपेयरिंग एट अबाउट 12 आवर्स पोस्ट इंजेक्शन एंड अटैक पीक नंबर बिटवीन 24 to 48 आवर्स। फिर इसके बाद में मोनोसाइट अपेयर होना शुरू होते हैं जो कि 12 आवर के बाद में इंजेक्शन के साइट में बढ़ते हैं, 24 से 48 आवर तक पीक पर होते हैं। मोनोसाइट आर अट्रैक्टेड टू द एरिया ऑफ रिएक्शन बाय मोनोसाइट कीमोटैक्टिक एंड एक्टिवेटिंग फैक्टर, इसको हम कहते हैं MCAF, मोनोसाइट कीमोटैक्टिक एंड एक्टिवेटिंग फैक्टर, एंड दूसरा इंटरफेरॉन गामा। इन दोनों के कारण मोनोसाइट का अट्रैक्शन होता है। TDTS cells also secrete another chemokines, the macrophages migration inhibition factor MIF, है ना, that prevent macrophages to migrate from the reaction site. तो रिएक्शन साइट से भागने के लिए रोकना हो तो TDTH क्या है, एक दूसरा टाइप का सिक्रेशन प्रोड्यूस करते हैं जिसको मैक्रोफेज माइग्रेशन इनहिबिशन फैक्टर कहते हैं। दिस मैक्रोफेज आर फर्दर एक्टिवेटेड बाय TDTH derived interferon gamma and tumor necrotic factor beta, which in turn activate more T cells. Thus, this self-perpetuating cycle occurs during type 4 hypersensitivity. This response may turn out to be beneficial and protective, destroying foreign antigen or microorganisms, or become detrimental, causing extensive tissue damage. तो ये था मीडिएटर्स ऑफ DTH रिस्पॉन्स। अब आते हैं प्रोटेक्टिव एंड pathological role of DTH response. Humoral immune response is of little significance is offering protection of intracellular bacteria because of the inability of the antibodies to reach the bacteria. Type 4 hypersensitivity plays an important role in the host defense against such bacteria. Lytic enzyme and oxygen metabolites of activated macrophages that infiltrate at the site of reaction kills the bacteria harboring intracellular bacteria. Kills cells that harboring intracellular bacteria. Immunity to mycobacterial infection is a DTH response in which activated macrophages was of the organisms to form a granuloma. If the DTH response is appreciated, organisms are killed and infection eliminated. Protective role of DTH in the mycobacterial disease such as tuberculosis, John's disease and leprosy is well known. A strong DTH response is elicited in the animal with early lesion of tuberculosis and paratuberculosis. Similarly, persons with tuberculoid leprosy with less number of bacilli show a strong DTH response, and those with lepromatous leprosy shows poor response. तो जिसको ट्यूबरक्लोइड लेप्रोसी है, ऐसे पर्सन को विथ लेस नंबर ऑफ बैक्टीरिया हों तो उसको DTH रिस्पॉन्स ज़्यादा होगा, और दोस्त जिसको लेप्रोमेटस लेप्रोसी है उसको DTH का रिस्पॉन्स कम होगा। द सिग्निफिकेंस ऑफ हाइपरसेंसिटिविटी इन द पैथोजेनेसिस ऑफ ट्यूबरक्लोसिस वाज फर्स्ट डिमॉन्स्ट्रेटेड बाय कोच, which was later named as Koch's phenomenon. Generalized tuberculosis develop if a normal guinea pig is injected subcutaneously with tuberculous bacilli. At the site of inoculum, hard nodule is formed, which breaks down to form an ulcer. Guinea pig dies within 2 to 3 months. If, however, a tuberculous guinea pig is injected similarly, an acute response consisting of exudation and necrosis develops at the site of inoculation. देखो, इसमें बहुत अच्छी चीज़ है, जिसको जनरलाइज ट्यूबरक्लोसिस डेवलप हो जाती है इफ़ ए नॉर्मल गिनी पिग इज इंजेक्टेड सबक्यूटेनियसली विद ट्यूबरक्लोस बेसिलाई। ठीक है, आपने सबकट टीबी बेसिलाई लगाया तो उसको जनरलाइज ट्यूबरक्लोसिस हो जाएगी एट द इनोकुलम, वहाँ पर एक हार्ड नोडुल बनेगा और अल्सर भी हो जाएगा। अब जिस गिनी पिग को और वो गिनी पिग 2 से 3 महीने में मर भी जाएगा। इसके अलावा इसके विपरीत एक ट्यूबरकुलस गिनी पिग है जो इन्फेक्टेड है पहले से ही, सिमिलरली एन एक्यूट तो उसमें क्या है, एक एक्यूट रिस्पॉन्स कंसिडर ऑफ एन एक्सुडेशन होता है उसमें और नेक्रोसिस हो जाती है, बस इसके अलावा कुछ नहीं होगा। द नेक्रोटिक टिश्यू सून स्लफ एंड हील विद इन ए short period. There is no generalization of infection and bacteria are killed rapidly. This is due to DTH response operating in the sensitized guinea pig. Beneficial and harmful effect of hypersensitivity depends on the circumstances. So, hypersensitivity in the small number of bacilli results in tubercle formation that enhances the kill of organisms by activated macrophages. तो कम संख्या में होते हैं तो बेनिफिशियल है। दिस हेल्प्स इन प्रिवेंटिंग ऑफ री इन्फेक्शन एंड ऑल्सो डिसमिनेशन ऑफ द इन्फेक्शन। हाइपर सेंसिटिव रिस्पॉन्स टू द लार्ज नंबर ऑफ बेसिलाई रिजल्ट्स इनटू एक्सटेंसिव नेक्रोसिस एंड टिश्यू डैमेज। लिक्विफेक्शन कॉज्ड बाय लाइटिक एंजाइम ऑफ मैक्रोफेजेस is considered most harmful in the pathogenesis of tuberculosis. Mycobacteria resist intracellular destruction by the macrophages until T cell activate them. Mycobacteria resist intracellular destruction by the macrophages until T cell activate them. जब तक T cell activate नहीं करता मैक्रोफेज को तब तक मैक्रोफेज से इंट्रासेलुलर बैक्टीरिया मरता ही नहीं है. Bacteria being rich in poorly metabolizable wax are not easily removed and hence the presence of prolonged time causing more and more accumulation of macrophages. The macrophages in this location change into epithelial like cells known as epithelioid cell. तो जो मैक्रोफेज होती है ऐसी लोकेशन पे वो एपिथेलियल सेल से लोकेशन चेंज इनटू एपिथेलियल टू सेल लाइक सेल्स नॉन एज एपिथेलियोइड सेल्स कहलाते हैं. वहाँ पे एपिथेलॉइड सेल्स कहते हैं मैक्रोफेज को. एन एग्रीगेशन ऑफ एपिथेलॉइड सेल्स सराउंडेड बाय लिम्फोसाइट्स इज रिफर टू एज ग्रेनुलोमा. तो सबसे पहले बैक्टीरिया आ गया, फिर एपिथेलॉइड सेल्स आ गई, फिर उसके बाद ग्रेनुलोमा आ गया, लिम्फोसाइट आ गए, तो इसको हम ग्रेनुलोमा कहते हैं. सेवरल ऑफ मैक्रोफेजेस आर फ्यूज्ड टू फॉर्म गैंट सेल. जैसे बहुत सारे मैक्रोफेज फ्यूज हो जाते हैं उसको गैंट सेल कहते हैं, या फिर गैंट सेल को हम लेंगरहैंस टाइप सेल्स कहते हैं. अ कैरेक्टरिस्टिक फीचर ऑफ ट्यूबरक्लोसिस लीजन. Foreign body type Gantt cell can also be formed if the reaction is sustained. Granuloma may be surrounded by a thin rim of connective tissue under the influence of the platelet derived growth factor PDGF and transforming growth factor beta यानी कि TGF beta secreted from activated macrophage. अब macrophage खुद ही वहाँ पड़े को सिक्रेट करते रहते हैं, उसको हम कहते हैं PGDF और TGF. ठीक है, तो ये कनेक्टिव टिश्यू की बाहर लेयर बन जाती है. दिस टाइप ऑफ ग्रेनुलोमा इज ऑल्सो कॉल्ड एज हाइपर सेंसिटिविटी ग्रेनुलोमा. तो ये था अभी तक हमारा ट्यूबरकुलिन रिएक्शन. अब आते हैं जॉनिन रिएक्शन. एनिमल जैसे कि कैटल, शीप एंड गोट इन्फेक्टेड विद द माइकोबैक्टीरियम पेराट्यूबरकुलोसिस show DTH response following intradermal injection of Jonin PPD. The maximum reaction reaches after 48 hours of inoculation. Intravenous Jonin's test is sometimes used for diagnosis of Jonin's disease. John's disease. A rise of temperature to 1 degree or neutrophilia recorded after 6 hours of inoculation is suggestive of positive reaction. तो अगर आपने इंट्रावेनस दिया और न्यूट्रोफिलिया मिला और 1 डिग्री टेम्परेचर बढ़ा हुआ मिला तो आप समझ लें कि जॉन्स डिसीज है. अदर इंट्रासेलुलर पैथोजन शोइंग DTH. बिसाइड माइकोबैक्टीरियल इन्फेक्शन, पॉजिटिव DTH रिस्पॉन्स मे बी ऑब्जर्व्ड इन सेवरल इन्फेक्शियस डिसीजेस कॉज्ड बाय इंट्रासेलुलर पैथोजन्स. Microbial extract is prepared from those pathogens for intradermal skin testing. Brucellin prepared from Brucella abortus is sometimes used for the diagnosis of brucellosis
with some limitations. Culture filtrates of Pseudomonas meliae, known as meline, is used for skin testing in the horses suspected to be suffering from glanders. A number of modification of meline test has been evolved, but most commonly used method is the intradermopalpebral test. इंट्राडर्मोपाल्पेब्रल टेस्ट ये करते हैं ग्लैंडर में. In this test, meline is injected into the skin of lower eyelid. Positive reaction appears after 24 hours, is characterized by edematous swelling of eyelid, lacrimation, and ophthalmia. Certain fungal diseases such as histoplasmosis and coccidiomycosis can be diagnosed by skin testing using histoplasmin and coccidioidin respectively. Toxoplasmin is used for diagnosis of toxoplasmosis. Most of these tests are considered non-specific, and because of being in vivo method, they also induce antibody production which hinders serological monitoring. क्योंकि ये इसमें एंटीबॉडीज प्रोड्यूस होती है ना, फिर बाद में सीरोलॉजिकली मॉनिटरिंग करना इसमें कठिन हो जाता है. नेक्स्ट हम पढ़ेंगे एलर्जिक कॉन्टैक्ट डर्मेटाइटिस. Several chemical and other substances have been found to cause allergic dermatitis, which was mediated by type 4 hypersensitivity. This includes formaldehyde, trinitrophenol, dinitrochlorobenzene, picric acid, aniline dye, nickel, turpentine, various cosmetic and hair dye, poison oak, then poison eve, IVY, EV, etc. ये जितने भी होते हैं ये एलर्जिक डर्मेटाइटिस करते हैं. Most of these substances are small molecule that can complex with the skin protein when applied on the skin. These complexes are internalized by Langerhans cells, यानी कि antigen presenting cells, processed and along with the MHC class second molecule and presented to T cell. Subsequent exposure with the same chemical causes infiltration of large number of mononuclear cells in the dermis within 24 hours. Intraepithelial vesicle are produced as a result of killing of Langerhans cells containing the chemical protein complex by activated T cells. The skin lesions is highly pruritic and hence known as allergic contact dermatitis. Pathologists working with the formaldehyde develop allergic contact dermatitis. In dogs, allergic contact dermatitis of ear may occur following neomycin treatment of otitis externa of ventral abdomen, scrotum. यहाँ पर भी होता है डॉग में. Following contact with the carpet dye. The clinical finding in contact dermatitis include appearance of mild erythema, यानी कि red patches, to severe erythematous vesicle formation, which are intensively pruritic. Self-inflicted injury may leads to excoriation and ulceration. Secondary infection with Staphylococci often leads to pyoderma. Chronic exposure to these allergens causes hyperkeratosis, acanthosis, and dermal fibrosis of the skin. Large number of mononuclear cell infiltration and vesiculation of the epidermal layer characterize the microscopic lesions. Closed or open patch test, patch test employing suspected allergen is carried out for the identification of the offending chemical allergen. Now next is the detection of type 4 hypersensitivity. Technique used for the measurement of cell mediated immunity are also applied to detect type 4 hypersensitivity. The in vivo method are intradermal skin test and DNCB test. The classical example of intradermal skin test are tuberculin and Jonin test. Cell mediated immunity can also be roughly measured by applying sensitizing chemicals such as DNCB. ठीक है, DNCB क्या है, इसको फुल्ली आप पढ़ लेना क्या होता है. In vitro method are used to measure either proliferation of T cell in response to the antigen or their ability to produce cytokines. Lymphocytes proliferation assay is carried out by incubating purified peripheral blood mononuclear cells with the antigen, followed by measuring the uptake of incorporated titrated thymidine by dividing lymphocytes. The assay can also be performed using MTT dye. Interferon gamma assay is another in vitro method to measure cell-mediated immunity. In this test, PBMC or whole blood is incubated with the antigen, and after 70 to 96 hours, supernatant is collected for estimation of interferon gamma. Estimation of interferon gamma is carried out either by bioassay or preferably by sandwich ELISA using MAB. The advantage of this test is that it does not interfere with the immune status of the animal. Thus, animal can be tested any number of times. Interferon gamma based sandwich ELISA are being used currently for diagnosis of tuberculosis and paratuberculosis in the cattle. So thanks for listening this chapter number 3, hypersensitivity, from book Veterinary Immunopathology Theory and Practice by Dr. R.S. Chauhan and B.N. Tripathi.